Good morning, everyone. So, what was the discussion in the previous lecture? So, kindly update me. I think we were discussing about cloning. Okay, we were discussing about cloning. Today, some remaining parts, those left now, I will try to finish it. Okay. Some factual data of cloning I already have given you, like first clone that was developed. Okay, in during the discussion of basic parts. Okay, I told you that first clone was those developed. It was a frog, an animal, as an animal. But the first in the animal mammal category was a dolly sheep. Okay, and those dolly sheep, clone was developed. It was a female clone. Okay, and one more discussion I told you in the basic parts of biotechnology that it was a mammary gland cell that was used. Okay, so it was a mammary gland cell, and you people know very well mammary gland is what. This is also this is a kind of somatic cell. So mammary gland cell was used. Later on, I told you like one of the clone it was developed in India. Okay, one of the clone was developed in India. It was NDRI, National Dairy Research Institute. Okay, it was the first clone of mammals. Okay, it was the first clone of animal. Okay, I hope you know very well that different facts I'm telling you. The first clone of animal was frog. First mammal clone was dolly. It was a sheep. Okay, it was a sheep. Okay, DOLLY. And the first clone in India, it was developed by NDRI, National Dairy Research Institute. I hope you know, because I discussed with you earlier, National Dairy Research Institute. So National Dairy Research Institute. So NDRI, National Dairy Research Institute. This is in Karnal. So this is in Karnal, Haryana. So this is in Karnal, Haryana. Okay, Karnal, Haryana. So these are the various parts in the case of cloning. Now the question is, okay, what about the uses of cloning? Okay, what about the uses of cloning? Overall, positive and negatives both. Okay, those cloning discussion I have done in the previous lecture. Those cloning discussion I have discussed. Here, what about its pros and what about its cons? What about its pros and what about its cons? You must be clear. So, what about the pros of cloning and what about the cons of cloning? Okay, that is important. So, pros of cloning is what? We can produce the same desired character. Okay, we can produce the same desired character. Here, same desired character we can produce. Okay, you know very well as genotype and phenotype both are same. As genotype and phenotype both are same. So, same gene and same expressions. Okay, you people want to know, sir, where same desired characters? Suppose guys, I just seen like some of the specific breed of cattle. It's giving a 50 liter milk in a day. So that breed of cattle's cloning if you'll do, so it's good. The same desired characters you can find out. Okay, if any of the plants have a better in diversity, okay, so that cloning if you'll do, so ultimately multiple number of plants, the same characteristics you can get it, because clonings are those, it is a kind of vegetative type. Okay, last day I told you it is a type of vegetative type. So definitely the same desired character produce, and same desired character produce the same desired multiple copy if you want to produce. So it's the best option. Okay, another one, we can save characters. We can save same characters. We can save same characters. So same desired character, okay, and save character means if suppose someone is going to lost, so that number we can, that organism we can save again and again and again and again. But there is one negative, guys. If same save characters you will do as same desired character produced, so finally you can save it. So there is one negative, guys. What about the negative? If same multiple copy of organism we will produce, so there is no variations. So because of cloning will not due to this, guys, have a no variations. Cloning not provide variations, and here Darwin said for evolution variation is important, and that is the reason if variation stop, finally what happened? No competition. If there is a no variation, so there is no competition. All desired organisms are same. If all desired organisms are same, so it's the same desired character. So no variation, no competition, and no variation, no competition. So ultimately, what will come the result? Okay, no evolution. So there is a reason, guys. In the coming future, there will be no evolution, and organism may stop here. Okay, organism may stop here. Second point. And Darwin said for evolution, variation is very important. Just like you people, guys. You all people are different in characteristics, and that's why you people involved in the competitions. And those guys will be better that people will select in the examinations. Because of variations, you people lead the competitions, and competition, because of it, better character evolutions. Okay, better character evolve, that is a evolution. Okay, even guys, one day a guy asked me, so if some of the organisms number are going to decrease, they are coming under the category of critically endangered type. So why we are not going further on clonings and save the numbers, increase the multiple copy? See, if same number you will produce, ultimately the animal is same. There is not genetic variations. Because same gene you are going to multiply. Okay, and this is the reason this all the animal will call as a one type of genotypes. Okay, if genetic variation come, in that cases I will tell you like this is a 100 type of animal, 100 in numbers. Your same genotype characters you are going to produce, multiple copy. So finally it is not a conservation of organisms, guys. You are wrong. Okay, because everywhere have a same gene. If suppose one of the gene have not the resistance against any problem, all the genes, every animals will die. Okay, because all have a same genotype characteristics. Okay, so you don't think so through the help of cloning animal or plants, those are going to critically endangered, we can produce the numbers and we can save it? No, it will all be a same genetic character, so it will again call as a one organisms, not more than one. If you want to produce more than one organism, so you must have to produce genetic variations. Okay, you must have to produce genetic variation, nothing more. Okay, everyone, fine. So this is here. So no variation, no competition, and no evolution. The first point. Okay. Second one, guys. What about the cons? Even in the case of human being. Okay, human being, complete cloning is banned. Okay, in the case of human being, if you will go for the complete cloning, then in the case of human, complete cloning is banned. Reason? Human is a social animal. Human is a social animal. We have our relations. Father, mother, brother, sister. Okay, son, daughter. We have a relation, guys. If suppose I am just producing some of the organisms, multiple copy of human being. Right now you are 20 years old and your baby is a 1 year old. You don't think so when your cloning will form exactly the cloning will be at 20 years old. No. It will be one day. And one day cloning or your cloning. Ab just you tell me what your mama papa will tell. My baby born again. Yeah, what you will tell? Me myself born again. Okay, yeah, you have a husband, your wife, or something is. Suppose Rajiv Gandhi clone I will develop. Suppose, don't take it other way. So, what does Sonia ji will do? Sonia ji will take that clone in their own hand and she is my husband. One day husband. I hope you can understand, guys. Even some other thing, guys. So in the case of human being, because human is a social animal, so totally, this cloning is not allowed. Okay, fine. So this is the human being, social animal. Next point. But in other cases, other animals, that is allowed. In other animal it is allowed. Only in the case of human being, it is not allowed because of social animals. Human is a social animal. Another one, guys. Therapeutic cloning is allowed in the case of human being. I will tell you, therapeutic cloning is allowed. Therapeutic cloning is allowed. Why therapeutic cloning is allowed? You people know very well. Same desired organ clones are going to be performed. If same organ clone happens, so no need of any other person's organ, and there is no chance of organ rejection. Last day I told you, guys. Okay, in the case of organ formation, okay, organ requirement, I will take organ of any of you people, so that will result inside my body, and that is the reason I have a need of immunosuppression medicine. Neither your organ will result inside my body, I can die. And there is a chances, guys, more chances. It's a risk. It's very risky. So that is the reason, if through the help of therapeutic cloning, I will develop my own kidney, I will develop my own cells, and I will take it inside my body. So chances of rejection are no. Immunosuppression medicines are not required because 100% it's your organ. Okay, fine. So therapeutic cloning is allowed in the case of human being. Okay, but complete cloning is totally banned. So which cloning I'm telling you in the case of human being is banned? Complete cloning is banned. Complete cloning is banned. Somebody's are mentioning like crime etc. can increase. One of the original organisms. Second is it's a copy organisms. If one will be the original organism and second will be its copy organisms. So you people can understand very well, guys. The copy organisms can go for the crime, but the other person will not do. So finally we'll have a confusion and doubts, and there is a chances to have a means this type of crimes can also increase. Okay, crime can also increase. Even through the help of cloning, people can go for the violation of body organs. Okay, people can go for the violation of body organs as a business profit and something other. Okay, so this is the overall here about some positive and some negatives. Okay. Manjushri, therapeutic cloning take some time. So after getting successful completely, even in those nutrient media, we are putting the stem cell for the development of different organs. It's a time consuming process. And another one, sorry, not time consuming. Actually, those nutrients we people have used for the development of organ, it's not properly developed. But yes, very soon this thing, even concept wise happen. Some organ wise we have also developed, but finally it could not achieve. So in the coming future, definitely whenever this technology will be very successful, yes, lot of the things even achieved. Lot of things even can achieve. Okay, everyone. So this is a characteristics here. You people remember nothing more in the case of cloning. So the factual part and conceptual part I have given you. Now come next question topic, that is called stem cell. That is very popular, guys. Stem cells. Stem cells. Even one feature of a stem cell you already have seen. One feature of a stem cell you people already have seen. Where one feature of a stem cell you have seen? I hope you know very well. Okay, one feature of a stem cell you people have seen in the case of therapeutic cloning, that from the blastocyst cell we are going to separate the stem cell. I hope last day lecture you did not miss, so you people know very well. So from the blastocyst cell you will just separate the stem cell, but you know there is a one negative here in the case of stem cell research. When you people are going to take a stem cell from the blastocyst, so this blastocyst is going to die. Where hardly blastocyst of 128 to 256 cell stage. Okay, 128 to 256 cell stage. Here few of the stem cell you will remove, so ultimately the whole embryo can die. Okay, this whole embryo can destroy or die. So this is the problem. Okay, this is the problem here in this case. Okay, fine. So stem cell have some controversy also. This is the reason I'm telling you. Some human rights are also against the stem cell, but better you people avoid these people. Okay, whenever these people have a need of kidney or anything else, okay, so finally they will go for the stem cell. Okay, in that cases they will be ready for it. Okay, the people have a lack of knowledge. Okay, and another one, guys, no rejections and everything you people know very well in this case. And here in this case stem cell, when we are going to take a stem cell from the blastocyst, in that period of time blastocyst is not the living conditions. Okay, even up to 5-6 month तक बच्चे तक abortion allowed होते हैं. Okay, but I'm not telling for abortion or anything, but I'm telling you just for 2 दिन 3 दिन 4 दिन में this blastocyst that form. I hope you know very well blastocyst how much days it form. Hardly 3 to 4 days. Okay, it's not a living organism, doctor is saying, because heartbeats are not coming, lot of things not there. So how you can say it's a living organisms? Okay, so there are few debates with the stem cell research also, and some debates those are here in the stem cell also. I describe you. Okay, I describe you which kind of debates here. Okay, everyone. So which kind of debates here? I hope you people understood. Now come next. Stem cell. So how to describe a stem cell? First of all, you come there. Okay, how to describe a stem cell? Okay, so it's in a very simple way. I'm just going to define you about the stem cell. Many properties, different types of stem cell I'll discuss. Okay, different types of stem cell I'll discuss. And after this class, you will not have any single confusion of the stem cell. Stem cell was a prelims question, and even last year it was a mains question also. How the stem cell can be used against the leukemia and Parkinson type of disease. It was a UPSC questions. Okay, so that all the entire discussion I'll do here. Okay, all entire discussion I will do here. Okay, now listen. How is stem cell defined? Okay, how is stem cell defined? Okay, how is stem cell defined? See, guys, a stem cell is what? Undifferentiated It is undifferentiated and unspecialized cell. So how to define stem cell? It is an undifferentiated and unspecialized cell. So a stem cell is what? It is an undifferentiated and unspecialized cell. Okay, 1 point. And this cell, those are undifferentiated and unspecialized, have tendency. Tendency means I can tell you have a property. Have a tendency or property to be differentiate and specialized. To be differentiate and specialized. Differentiate and specialized. So a cell has no differentiation and no specialization in the beginning, but have property to be differentiated and specialized. And I hope you can easily understand, guys, when we put a stem cell in the different nutrient media, stem cell as undifferentiated, but whenever you have put into the nutrient media, they develop as a different organs. Somewhere has a kidney, somewhere has skin formation, something else. So what you got it? Definitely an undifferentiated and a specialized cell have tendency to be differentiated and specialized. It's a very unique property of the stem cell. Okay, it's very unique property of stem cell, you people remember. Okay, so it's very unique property of stem cell. So an undifferentiated and specialized cell being a tendency to be differentiated and specialized. Okay, differentiated and specialized. Now you come into the different types of stem cells. We are coming here. Okay, different types of stem cell. Different types of stem cells. So different types of stem cells. What about the different types of stem cell? See guys, the one that is called totipotent. It's a technical term, but I will simplify the things. One is the totipotent. Second is pluripotent. And third is multipotent. And third is a multipotent. So one is totipotent, second is the pluripotent, and third is a multipotent. So totipotent, pluripotent, and multipotent. So these are the three types of stem cell: totipotent, pluripotent, and multipotent. What are these? See guys. And yes, all the cells have in different places, guys. Okay, all the cells have a different places. Okay, all the cells have a different places. So totipotent, pluripotent, and multipotent. See, what about the totipotent? First of the property of totipotent. Totipotent nature of stem cell are those cells where each and every cell have ability to development of entire organisms. Try to understand. A single cell have ability. I will simplify everything. Trust me, guys. Right now it's looking like a fact. After 10 minutes will begin as a concept, and you will not have a single confusion in the case of a stem cell. A single cell can form entire organisms. Entire organisms. So totipotent is what? Where a single cell can develop entire organisms. Totipotent is what? Where a single cell can develop entire organisms. So totipotent means if a single cell have ability to development of entire organisms, this is what? This is a totipotent. Okay, what is a pluripotent? Pluripotent, if a single cell have ability to develop different organs. A single cell have ability to develop different organs. A single cell have ability to develop different organs. Okay. Different means I'm here telling you better is many organs. Not better, different. You people use a term that is called many organs. For the development of many organs. Many organs. Second case, pluripotent. A single cell have ability for the development of many organs. This is a pluripotent. What about the multipotent? See, a single cell which cell? Stem cell, a single stem cell I'm telling you. Okay, on that basis of property there is different type: totipotent, pluripotent, multipotent. Okay, multipotent means a single cell have ability for the development of few organs. Few organs. Few organs means very, very rare. Very few guys. Very few. Very few. So I hope you people very clear here in this case. Okay, I hope you people very clear here in this case. So totipotent nature of stem cell. See, it's a nature of stem cell. Actually, inside your body, those stem cell present. One point, it's present in different places inside your body. And second one, at the time of your phase of development, means when a baby is going to develop into the mother body, until now, in different phases of your lifespan, okay, have a stem cell of different properties. Throughout your lifespan, guys, different places have a stem cell with have a different properties. Yes, and that I will prove. Okay, everyone. So I hope you people very clear here. Nobody have any doubt in this case. Just a minute. Now, totipotent, pluripotent, multipotent. You just come here, guys. What will come here? Actually, if you people have seen a zygote, you know zygote. Last day I showed you guys last time. I shown you the zygote. Cells from early embryos. Here the answer is a cells from early embryos. Cells from early embryos. So cells from early embryos. And these embryos are hardly 1 to 3 days of embryo. Okay, 1 to 3 days of embryos. Okay, 1 to 3 days of embryos. 1 to 3 days means marula. I hope you understood. Marula around 1-2 days up to marula. Okay, and just tell me, guys, each zygote cell have ability for the development of complete organisms or not? Yes. Zygote have ability for the development of complete body or not? Yes. After zygote, if cell divided and become 2, each and every cell, if it try to develop a baby, so they can develop a baby. Yes or no? Yes. Yeah, 2 become 4 and each and every 4 cell become as a separate for the formation of entire organism. Hey, I have discussed in the case of twins. Do you remember identical? Do you remember identical twins? 1 cell, yeah, 2 cell, yeah, 4 cell. They have ability for the development of complete organisms. And this type of property of stem cell is called as a totipotent nature. Just tell me, guys, you people clear with the concept of totipotent or not? What about the totipotent cell? See, zygote is that kind of stem cell. Zygote people also consider as a stem cell type property because it has ability for the development of complete body. So 1 day or 2 day, okay, and when the zygote divided from 2 cell, each and every cell it become free and they all develop the body. So 2 baby can develop, entire organism can develop. Entire organism means complete body can develop. Yeah, four cells form. Okay, mitosis. I hope you know very well. During the development of embryonic development, see, in the first lecture of biotechnology, I describe every concepts. Okay, last day I have, this is the reason I have discussed what happened in the case of double fertilization. I want everyone answer yes. Arey wow, people already have given me the answer. Yes, sir. Monozygote, sir, everything. And see, here those cell present, na, this is also a stem cell type of properties. And this type of stem cell of properties is called as a totipotent nature of cells. I hope everybody clear. Okay, what about the pluripotent? So here what is it? Cells from early embryos. Pluripotent nature of stem cell you will get into the blastocyst. I hope you remember last day. Very good, very good, very good. Everyone has given me the one. Great. Okay, some cell of blastocyst. Here the answer is some cell of blastocyst. Some cell of blastocyst. So some cell of blastocyst. Some cell of blastocyst, it's a pluripotent type. So some cell of blastocyst, okay, some cell of blastocyst, it's a pluripotent type. Okay, inner cell mass, this is also called as a inner cell mass. Inner cell mass. And this property, I hope you remember last day you have seen in the case of therapeutic cloning. Tell me, guys, this property you people have seen during the case of therapeutic cloning or not? We have taken the sum of the cell from the blastocyst, I hope you remember. And we have put it into the various nutrient media. In various nutrient media, various type of organ formation. But many organs formation happen. So many organ formation happen. So you tell me the second one. You people clear? If you clear, so tell me yes, sir, clear. Okay, write clear. Okay, because sometimes you people write yes, sometimes you people write one. Right, clear. So those last day discussion, last day means Saturday, those discussion I have done, I hope you people know very well what was there. A single cell of blastocyst have ability, okay, a single cell of blastocyst, okay, blastocyst, you know, blastula stage, okay, and the stem cell have ability for the development of many organs, different types of organ, and that become possible. And what is the blastocyst? It is also called as a ICM, inner cell mass. Great, great, great. These all the clear and all the yes sir, those you are telling me na, these all the benefit because of basics. These all benefit because of basics. Okay, so inner cell mass, ICM. ICM. So I hope you people very clear. It's about inner cell mass. This is called ICM. What about the multipotent? Multipotent you will get into the adult stem cell. Okay, into the adult stem cell. This is in the adult stem cell. This is actually in the case of adult stem cell. This time inside your cards. Okay, yeah, backside. You know, those stem cells present inside your bone marrow. Okay, that is what, that is the adult stem cell. And here the stem cell have ability for the development only few organs. This is the reason this type of stem cell is not the best stem cell. Best stem cell if you want, so that best is this blastocyst vala for the experiment purposes. Okay, the best is what? Okay, this blastocyst vala, this is the best one in this case. Okay. So this is the overall here in this case. And yes, one thing, even when a baby is going to born, you know, when a baby is going to born, cord blood cells and all. Okay, cord blood cells, cord bloods. Okay, cord blood cells and all. What the doctor is going to preserve? Okay, their own stem cells. Their own stem cells. And what about the procedures used for the preservation of the stem cell? That's called cryopreservation. Generally, it is the cryopreservation. It's a very common procedure. Cryopreservation technology used for the preservation of stem cell. Okay, for the preservation of stem cell. Cryopreservations. Well, cryo means low temperature based technology. At minus 196 degrees Celsius temperature in liquid nitrogen. In liquid nitrogen, stem cells, genomes, tissues. This is safe. This is safe. How it is safe? This is safe in the living form. This is safe in the living form, but this is inactive. But this is the inactive. So those stem cell we are using and put into the cryopreservation method for its safety and conservation. So this is what? This is inactive, but in living form it is safe for 20-30 years. But whenever you want to develop, okay, living will be already there, just you make it active form. And when you people will make a stem cell active, further the stem cell will develop entire organism of you people guys. Even if a baby is going to born, many of the companies they are working. Cord lice, many of the companies, okay, I'm not going to advertise any companies. Okay, when the babies are going to born, that umbilical cord is cutting. That umbilical cord cut. And when the umbilical cord cut, guys, inside have a stem cells. And this stem cell, okay, the people are going to preserve under the cryopreservation. This is the procedure those we are adopting. Okay, that we are using. And in that way, guys, for long duration of time, 20 years, 30 years, after it a baby have need of any organs, anything else. So the stem cell can be used for many therapies. Okay, the stem cell can be used for many therapies. What about the pros? What about the cons? I discussed with you and everybody know what are here in this case. Okay, so I hope nobody have any doubt here in this case. I just to simplify every concepts. Okay, another one. You just see. Actually, this stem cell. This is stem cell, you just see. This is stem cell, those we can see here as a totipotent type. Totipotent and pluripotent. These are called as an embryonic stem cell. These are called as an embryonic stem cell. And this is what? This is the adult stem cell. This is the adult stem cell. So this is what? This is the embryonic stem cell, and this is what? This is the adult stem cell. So embryonic stem cell and adult stem cell, you people remember. Okay, this is the embryonic, these two. So if I ask you, yeah, UPSC ask you, okay, what about the nature of embryonic stem cell? The answer is the totipotent and pluripotent. What about the nature of adult stem cell? You will say as a, that is a multipotent nature. Multipotent nature. Okay, fine. Actually, you know, guys, against many kind of diseases, against even many therapies, okay, many therapies, stem cell is the best option. Stem cell is the best. Okay, in different therapies, stem cell is the best. And now come here into the application parts. As UPSC has in your paper, okay, as UPSC has in your paper, guys, okay. Even the application of stem cell. Even application of stem cell. Application of stem cell. So write down about the name of disease like cancer, type 1 diabetes, cancer. Okay, write down. Next one, Parkinson disease. Later on you write that is the arthritis. Even many of the diseases, but few of them I'm just writing here. Okay, not more. Cardiac failure, tissues वगैरह damage and all. Okay, cardiac failure. Skin burning if you happen. Skin burning. You know, many of the applications, etc. Many of the applications are possible. Skin burning, many of the problems. Okay, stem cells even also used for the new drugs testing and faster development of drugs. For new drug testing, it can also use new drug testing and faster development of drugs. Okay, many of the applications. Few of the beautiful application I'll discuss with you. Okay. Cord blood cells is umbilical cord. Okay, when the baby's umbilical cord is going to cut. Okay, cord blood cell is what? When the umbilical cord is going to cut. Okay, that is the cord blood cells. When a baby born, what happened? That umbilical cord is going to cut. Okay, in the beginning it was called placenta. Later on it is called umbilical cords. Okay. So cord blood cells. When the umbilical cord cut, here some of the tissues doctor even preserve in this way. Okay, but one more is the backside wala. But adult stem cell is a backside wala. जो bone marrow से we are going to separate, that is umbilical cord blood cells. Two places we are using it. Okay, but one thing I will clarify here. In the case of umbilical cord wala, when it is cutting. Okay, in that time of birth, those stem cell we are going to preserve. It has a very best nature. Okay, very best nature, and those we are taking from the backside of the stem cell. Okay, bone marrow वगैरह से we are going to separate. Okay, it is not better, and this is here in the case of multipotent nature. But umbilical cord wala that we are going to cut now, it is a embryonic type, it is good. So generally those cord blood cell I have mentioned in the case of multipotent type, this is the backbone wala. Backbone inside those cells are there. It's not the umbilical cord wala, because umbilical cord wala is better. It is a pluripotent nature. Okay, it is a pluripotent nature. Neither only few organ you will develop from the umbilical cord wala, this wala. So card two places we are going to use. Okay, so here don't be confused. Two types of cards I'm telling you. One is the best, second is not best. In the case of adult stem cell, I'm taking from the backbone के side से जो card है ना, spinal cord वगैरह. The card is represented as a spinal cord वगैरह back side वाला. It's a multipotent nature. Dialysis patient not go for the stem cell therapy. Okay, but dialysis patient the kidney is not having effective. So kidney you can develop through the help of stem cell. Okay, kidney you people can develop through the help of stem cell. So this is the point. You people remember. Okay, now you just listen, guys. Some of the information I want to give you in some better way about the application of stem cell. See, even stem cell is a very big topic. I can take the whole day lectures of it. But according to UPSC, what about the requirement I'm discussing here? Okay. Suppose some of the tissue even getting weak. Some of the tissue getting weak. Yeah, some of the tissue even got damage. I can tell you, like in the case of type 1 diabetes, like in the case of Parkinson disease, arthritis, cardiac failure, skin burning, okay, this type of even cancer, everywhere, this type of features stem cell can achieve. This type of feature stem cell can achieve. How it is so? Very simple. First you just recall what about the property of stem cell I told you. Sir, it's an undifferentiated and a specialized cell, and the stem cell has the property to be differentiated and specialized. I hope you remember. An undifferentiated and a specialized cell have property to be differentiated and specialized. This was a property everybody know. So I'm here telling you guys. Okay, अब just like a person is suffering with the leukemia, because it was a mains question paper in your examination. Leukemia. What is the leukemia? Leukemia is a case of blood cancer. Leukemia is a case of blood cancer. So what is leukemia? Leukemia is a case of blood cancer. Why you people have a blood cancer? See guys, first of all you must be clear. Bone marrow, I hope you people know very well. Which portion of your body is responsible for the formation of your blood? Bone marrow. Bone marrow is a soft tissue which are present inside the bone, guys. Bone marrow. Okay, bone marrow is responsible for the formation of your blood cells. Okay, bone marrow is responsible for the formation of your blood cell. Even one of the questions many years back, 15-20 years back, UPSC asked, in embryonic condition, just a information I'm telling you, in embryonic condition our blood was synthesized by liver, but by birth our blood synthesized by bone marrow. It means when we people were inside our mother body, so what happened? Firstly our liver formed, and then from our liver there was a formation of blood. But when we people born, later on what happened? Liver stopped to do this mechanism, and now this role is going to play by bone marrow. In this time, which portion of your body cell is responsible for the formation of your blood? The answer is the bone marrow. Me, myself and you yourself, both. Me, myself and you yourself, both. Now just tell me guys, blood cell form means RBC, WBC, platelets, everything is going to form from the bone marrow. What happened in the case of leukemia? In the case of leukemia, number of WBC abruptly increased. Those WBC inside your blood, 1 ml blood, there's around 5,000 to 12,000 at WBC. It number increases lakh and lakhs. Okay, and then through the help of it is being identified that you are suffering with the blood cancer. So what happened in the case of blood cancer? Blood cancer is called as a leukemia. WBC number abruptly rise. And why it happened? Because of the abnormality of bone marrow. Because bone marrow is the tissues which are responsible for the formation of blood cells. Here WBC number abruptly rise, and now it's a case of blood cancer. What he will do? Nothing guys. You just inject the bone marrow cells. Again I repeat, you just inject the bone marrow cells. Where? A stem cell in the bone marrow. You just inject the stem cell into the bone marrow, and those problems happen to the bone marrow. All recovery why? Because the stem cell have ability to differentiate into and specialize into different body parts. So definitely those recoveries were important. Here the stem cell start to form the normal bone marrow. And those defective bone marrows there slowly gradually they will be replaced by the normal bone marrow, and there is a possibility that person can treat against the blood cancer. Against the blood cancer. Suppose in the case of cardiac diseases, some other diseases, by birth some tissues, kabhi you people heard it na, the heart have a pores. Impure blood is going inside your body, and that is the reason body has not proper stamina, because impure blood is moving, and that is the reason the people can die. Okay, something this type of news you will get it guys. Maybe any of family members and nearby any of known people you will get it. If that type of pores are anything here, so here we can also inject the stem cells, and inject stem cell can do the recovery of pores those are present inside your heart. Possible. Even long back, 8-10 years back, one of my students called me and just asked me, sir, I'm suffering with the one of our eyesight problems. Okay, doctor said my eye is fine, but backside of tissue is damaged, and uske backside everything is fine. So ultimately the front side of eye and the backside tissue just damaged, and backside further everything is fine. So in between have a tissue damage. So doctor has given me one hope about the stem cell therapy. Through the help of a stem cell, if you enter there, the stem cell will further differentiate and form a specialized tissues, that is your backside though tissue damage, and further there is a possibility to have a eyesight that will come inside you. So this is these are some features, even see guys. This is the type 1 diabetes. Type 1 diabetes is what? Type 1 diabetes, in this way UPSC never ask you to go and define the diseases and treat the stem cell. I'm just to tell you how the stem cell can be used as just informations. Okay, this dictation is not possible. Only try to understand it. Type 1 diabetes is what? Type 1 diabetes is called insulin dependent diabetes. Type 1 diabetes is what? This is a insulin dependent diabetes. This is the insulin dependent diabetes. What is insulin dependent diabetes? Actually inside your pancreas have a very specific cell that is called beta cell. It is present inside the pancreas. I hope you heard it, pancreas. This is one of the organ. Okay, this pancreas have a beta cell, and this beta cell is actually responsible for the formation of insulin. Okay, if insulin formation is not going to be happen, It means some tissues abnormality happen. So definitely stem cell will do the treatment of this cells. Okay, beta cells and all problem those happen in the pancreas, and now the people start to form the insulin. Naturally, insulin formation start because of some abnormality in the beta cell type of tissues. Okay, so tissue development, recovery, many type of treatments. Okay, organ you can develop in the case of therapeutic cloning. Okay, in the case of many application, now you people got it. Ab you people tell me, you people 100% clear with the many applications. As according to UPSC, those was required, I discussed with you guys. You people tell me, you people 100% clear. Okay, what about the debate? What about the debate with the stem cell? You know very well. Okay, that people are saying that is the kind of violations. Okay, we are going to kill an embryo and all. Please don't think like this. Okay, I hope entire concepts I simplify in very brief, but I have given you the many of the examples. Some of the examples those I have seen in my life. Okay, and even one thing more, guys. Okay, even if your baby is going to born, you should preserve the stem cells of your baby, because during our period of time, this type of technology was not invented. Okay, and through the help of your baby's stem cell, that stem cell can be used for the therapy of whole family members. How whole family members? Up suppose you have two babies. Okay, first baby in that time of birth you could not preserve the stem cell, but second baby's time birth you people have preserved the stem cell. That second baby's stem cell you can use for the treatment of first baby and you parents also. Why? Because the first baby also from you parents and second baby also from the same parents. So those genetic character in the first baby at least similar it is in the second baby. The second baby stem cell can use for the treatment of first baby, and even if your parents have some issue and requirement, so that second baby stem cell can be used for the treatment of parents problems. Okay, so how much the important stem cell and its future? But everything I am telling you, it's on research level. A lot of achievements happen. Okay, and definitely you people should go for the preservation of the stem cell in the case of your newborn. Okay, किसी ना किसी का एक का तो you should preserve. Great. बहुत बढ़िया. Excellent. अब now I'll come into the one of the very interesting topic. Even it was a last year prelims question paper also, pro-nuclear wala issue. Okay, 2020 it was a question in the case of pro-nuclear. So now come here. Three parents baby. This is also called as a pro-nuclear technology. And one more term is used there, that is called mitochondrial gene therapy. This is also called as a mitochondrial gene therapy. This is also called as a mitochondrial gene therapy. Cryopreservation. Cryopreservation, I define you. What is it? So three parents baby a mitochondrial gene therapy. Okay, so three parents baby and mitochondrial gene therapy. So three parents baby and mitochondrial gene therapy. See guys, you just recall the basic parts. Where is the DNA? I have a questions. 2 times UPSC asked these prelims questions. Where DNA available in a cell? This is my questions. Where is DNA inside the cell? Give me answer. You have your chat box. Connect me. Where is the DNA inside the cell? Where is the DNA inside the cell? And it was a UPSC question. It was a UPSC question. Where is the DNA inside cell? So where is the DNA inside a cell? Right. Okay. A perfect answer I got from someone only, not from everyone. The correct answer was nucleus. Nucleus means chromosome, chalega. Mitochondria and chloroplast. Okay, nucleus, chromosomes. मतलब nucleus के अंदर already is in chromosome. So better nucleus or chromosome, any of one, you people take it. And the another one is mitochondria and chloroplast. These are the three places. These are overall 3 places inside the cell where the DNA present. Okay, where DNA present. Now come. Okay. It means, achha, where is the DNA in the human being? The answer is a nucleus and mitochondria. Then the answer is a nucleus and mitochondria. The answer is a nucleus and mitochondria. Okay, then answer is a nucleus and mitochondria. Achha, today onwards your class timing I hope you know very well, 8 to 10 and 10:15 to 12:15. Because my target to finish your syllabus in this week, maybe Friday or Saturday. Okay, and just somebody's told me, sir, better you can increase the length, even duration of time. So I have decided that class will be for 4 hours. But 2 hours after it, I'll give you 15 minutes break, not 5 minutes. Okay, then again for continue of the next 2 hours. So I think it will be better, and you people will give me proper time. As somebody's told me last day, sir, better you increase the durations and take proper time in these discussions. And I think you know very well, I'm taking proper time, with giving more proper time even to discuss the entire subjects. Okay, reason I know very well, you people have a hope with the teachers. Okay, and I want to fulfill it. 100% satisfaction never possible. It's a physics law. 100% energy not transfer. But at least the maximum people, 70%, 80%, they should have a confidence. Okay, that is very, very important. Fine. So nucleus and mitochondria. Okay, but one thing again I'm telling you, nucleus have a main genetic characters of your body. Nucleus is the main genetic character. Nucleus have a main genetic character of your body. So nucleus have a main genetic character of your body. So nucleus have main genetic character of your body. Okay, nucleus have main genetic character of your body. But apart of this, some other genes also available in mitochondria. But that is a some genes. Some genes. Mostly genes are here, guys. Mostly genes are here. Okay, you people are developed because of nuclear genes, you people remember. Okay, you all people develop because of nuclear gene, not other. Okay, not other, you must be clear. Okay, fine. One more thing, guys, one more thing. Suppose, okay, I'll tell you one disease that is called Leigh syndrome. Leigh. It's a called disease. Syndrome is not actually disease, it is a kind of abnormality. I'm giving you a background of Leigh syndrome. Scientists identify if a baby is suffering with the Leigh syndrome, so here the reason is the mitochondria. Those genes available in the mitochondria, this is responsible for this type of disease. This is responsible for this type of disease. I directly enter into the information, guys. Don't like to waste my time. A syndrome was identified. Those syndromes are abnormalities going from generation to generation through the help of mitochondrial DNA or mitochondrial gene. Now I have one questions, and I hope everybody will give me satisfactory answer. Reason it's basic part I discussed. My question is, we people got mitochondria. We all people got mitochondria. So we all people got mitochondria at the time of reproduction from our father or from our mother. Give me answer. From our father or from our mother? Tell me. We all people got mitochondria from our father or from our mother? I have discussed these concepts earlier, guys, because maternal nutrition wala concept I have discussed. We all people got mitochondria from our father and mother. I hope you recall, guys, about the zygote. Okay, what are inside the zygote? I hope you know very well. Father nucleus, mama nucleus. Okay, but mama cell organelles. Entire mama cell organelles. I hope you remember. Entire mama cell organelles. Everybody, right? Cell organelles we got from mother. Absolutely right. Absolutely right. Okay, fine. Now tell me. It means those mitochondrial defective genes coming inside your body. Here, which one is responsible? Your mama is responsible. Your mama is responsible. Okay, because if mama have a defected mitochondria, so definitely you will have a defected mitochondrial expressions. Here, father is not responsible. Here, father is not responsible. So how this type of treatments we can do? Through the help of another lady. Those mitochondria is not defected. So how this 3 parents baby combination form? You just see here. Okay, you just see here. You all people have a correct answer. You just see here in this case. Okay, suppose this is a sperm. Suppose this is a sperm. Suppose this is the sperm. Sperm means own father, everybody know. This is what? Cell organelle. This is what? Nucleus. This is nucleus. And outside have a cell organelles. Sperm means, I hope you know, it's a father. It's a father. Second side, this is a mother. It's a mother. Okay. It's a mother. And it's a own mother, I'm telling you. It's a own mother. It's a own mother. Okay. Second one is the own mother. What is here in the own mother? See guys. It's a nucleus. What is here in this own mother? Have a nucleus. Okay. It's a nucleus. So own mother have what? It's a nucleus here. And what is here? What is here? What is here? Cell organelles. Cell organelles. Here I'm just marking it. This is the defected mitochondria. This is defected mitochondria. This is defected mitochondria. Okay, this is defected mitochondria. Okay, everyone, fine. Another side I will take another female, another female, another female. This is the egg. This is a nucleus. This is the nucleus. This is the nucleus. This is normal mitochondria. This is normal mitochondria. This is normal mitochondria. This is normal mitochondria. So it's a nucleus and it's a normal mitochondria. And here I'm telling you other mother, not own mother. It's other mother. This lady I'm also telling you as a mother, other mother. Other mother. Okay, now how these three parents baby formation happen? Now how these three parents baby formation happen? See guys, how the three baby formation happen? See here. See, what will be the next? This mother, from this mother. First of all, you people do the removal of nucleus. What you people will do? Removal of nucleus. First of all, you people do removal of nucleus. Because of other female chromosomes and genes are there. The removal of nucleus. If removal of nucleus you will do, what will come here? Those are the other mother. Other mother. In this other mother have what? Now normal mitochondria. Now normal mitochondria. Now normal mitochondria. They have what? Normal mitochondria. It's what? Normal mitochondria. Mitochondria. And this nucleus of own mother, you just separate here and put inside. Put inside. Put inside. So now what is this? Combinations. This is the combination of two mother. Now this cell become as a combination of two mother. या एक काम करो, रुक जाओ. This is the combination of what? Two mother. Two mother. What you people got in this two mother, you know very well. Normal mitochondria of another lady and own mama nucleus. Own mama nucleus is here. And now what happened? When there is a fusion of this sperm with this egg cell, it will be the result of three parent baby. It will be the result of three parent baby. Tell me. Everybody clear? I want everyone answer yes. I want everyone answer yes. How these three parents baby form? I hope you people know very well. I want everyone answer yes. And here I advise to all of you. You have a 1 minute, draw this picture. And if any doubt you have, so please ask me. You have only 1 minute, and then I'll go into some factual data. Okay, in a very simple way, I just to define you guys how 3 parents baby form. And finally, the removal of mitochondria, we have done it. Shubham, why not you have a present the stem cell in your backbone? Definitely it is present. But with time, guys, you know, the efficiency of stem cell and everything is going to decrease. Okay, efficiency is now slowly and gradually it is not being like to the normal type. Okay, normal type. The first 3 parents baby was born in Mexico. Mexico. The 3 parents baby was first baby already born in Mexico. Right now. And here Manjunath asked me a question. And I agree with Manjunath. This is only one debate in this case. Only one debate in this case. Manjunath is telling me like, sir, other mother's mitochondria has different DNA. Won't it be a problem? Yes, it may be a problem. It may be a problem. And this is the doubt only scientists have. Okay, but let us see in the coming future what type of problem this baby can face. There is a possibility. Definitely there is a possibility. But one treatment also happen, those mitochondrial defect happen. It will be in the coming future will come, but there is a few statements as a cons that there is a possibility in the coming future that other mother mitochondria can lead some problem into the baby. But you people know very well, 99% plus genes are in the nucleus, less than 1% are in the mitochondria. How much it will be effective or not effective, let us see. Okay, so there is a cons even suspect that there is a possibility this type of abnormalities may be there, but definitely this type of abnormality will come. Here you people cannot be sure, but scientists are believing it. There is a possibility. And see, this type of mitochondrial gene therapy, those I have done here. Okay, it's a part of also designer baby concepts. Okay, it's a concept of designer baby also. So it's a kind of designer baby also. Stuti, it is not so much easy task. It is not so much easy task. As it is achieved in European countries, it's fine because they are very good in the experiment and research and all. Even during the discussion of in vitro fertilizer and test tube baby, I told you. Okay, maximum success rate of in vitro clinics, you'll see overall it is in European countries. वैसे भी वहाँ बच्चे बहुत कम होते हैं और उनको भी अगर बचाया ना जाए तो फिर बच्चे रहेंगे कहाँ. इंडिया में स्टिल हैव अ 50% प्लस सक्सेस रेट, ज़्यादा ही है 60% आउट, बट यूरोपियन कंट्रीज मोर देन 70%. So if any of the family members are suffering with this kind of pregnancy, anything else, this problem, so you people should talk to the European countries. They are providing the better treatments. Okay, definitely those processes happen. It was in the laboratory. Okay, it was in the laboratory. Okay, it was in laboratory. Now you come into gene therapy. Okay, gene therapy. So we'll come into the gene therapy. Gene therapy also many times in the news. And just in the brief, I'll tell you what is a CRISPR-Cas9. Okay, because it's also the gene editing technology. But first of all, I'll discuss about the gene therapy. Okay, gene therapy. Actually, you tell me, do you know about micropropagations? 1 minute. Because at the same time, I should discuss about micropropagations. Micropropagation. Micropropagation. Tissue culture experiment is also called as a micropropagation. Tissue culture experiment is also called as a micropropagation. Okay, tissue culture experiment also called as a micropropagation. So tissue culture experiment also called as a micropropagation. Okay, so tissue culture experiment also called as a micropropagation. Also called as a micropropagation. What will come here? See guys, the one guys, those stem cell experiment you have seen, stem cell, those stem cell you people have seen, those stem cell you people have seen, it's a part of tissue culture. Those stem cell research you have seen, it was a part of tissue culture, you must be clear. So those stem cell research you people have seen, it was a part of stem cell. Oh sorry, is tissue culture, because a cell and tissues you are using for the development of various tissues or organs, and everything you people are using here in vitro methods. In vitro methods you are using artificial method. In vitro methods we have used it. The stem cell that you have seen, it was a kind of tissue culture experiment. I hope you understood. Oh, you people understood here. You people have not any doubts. Stem cell was what? It's a kind of tissue culture in the laboratory. Yeah, overall this technology also calls a micropropagation. An alternative name can UPSC can ask you, what is a micropropagations? And some statement of tissue culture stem cell are connecting together, so that is right. So animal wala tissue culture, so you have a seen, that's the best example. But I did not say the stem cell was a tissue culture experiment at least. Yeah, micropropagations. So here you people can be confused. But another example I will give you, though still I have not given you, in plants, tissue culture in the plants. Tissue culture in the plants. The animal wala, I hope everybody clear that stem cell was the best examples. Similarly, other we can also use it, but one example you must be clear. Plant tissue culture. I am coming directly into the plant tissue culture. Plant tissue culture. Actually, through the help of plant tissue culture, we can produce multiple copy of the plants in the laboratory. We can produce multiple copy of the plants in the laboratory. Okay, I'm giving you one methods, a tissue culture experiment of the plants, how we can do tissue culture in the plants. See guys, suppose I'm going to Japan, okay, and just I like some of the plants there. I like some plants there. Okay, and the plant is very big size. Okay, and through aeroplane, they will not allow. And if I want to take it delivery, again not allow. The government not allow. But I want to take it in my pocket. But I can take it in my pocket smartly. What I'll do, some explant I will use, the leaf explant. I suppose this is a leaf. Okay, I'll describe it. It's very interesting. You see, suppose it's a leaf. It's a leaf of the plants. Okay, it's a leaf of the plants. Some small explants. Explant means some parts of the leaf. Okay, that's called explant. Okay, I'm just taking it. Yeah, leaf. This leaf I'm going to carry from one place to another place, in my pocket, in my bag, anywhere, guys. Ab ye identify nahi ho payega. Okay, easily you can carry it. Okay, fine. If you are going in Europe, any of the plant leaf you are taking in your pocket, then anybody will arrest you? No, not arrest you. Okay, it's a general. You people have taken something, so something like it explant. But you know, this plant I will produce in India. In India, how? Tissue culture experiment. This explant have a tissues, cells. Yeah, they have a tissues, cells. I'll put this plant into some chemical medium. Lot of the chemical medium for the growth of plant tissues, they are developed. Morasus Coos medium, Noops medium, CJEC mediums, a lot of them. MS medium is very common. I'm writing is MS medium. If anybody is here from a tissue culture experiment and all they have done, so you know, acha chaha sir, I know very well. That's some chemical nutrient media. Designer baby is not a 3 parents baby. Okay, but 3 parents baby is a designer baby. It does not mean all designer babies are 3 parents baby type. Vikas. Okay. 3 parents baby is a type of designer baby. It does not mean all designer baby is a 3 parents baby. Other designer baby also there. So chemical medium, MS medium, Morasus medium. What happened in this chemical medium? It's a nutrient medium. It's a nutrient, guys. Nutrient means food. Nutrient means food. It's a nutrient medium. In this nutrient medium, what will come? This cell and tissue will divide and redivide, and they form a callus. They form callus. What is callus? Callus is an undifferentiated mass of T cells. Undifferentiated mass of cells. Mass of tissue. Undifferentiated mass of tissue develop. In this nutrient media, what happened? This explant, some tissues, those I have taken, I just put it. It will grow. It will grow. It will grow. And whenever it will come and form as a callus. Callus is what? Undifferentiated mass of cells. Yeah, undifferentiated mass of tissues. Undifferentiated means here I have a no formation of root, stem and leaf, that is called callus. Then what happened? Then what? I just put this callus into some hormones. Hormones means those are nutrients, very specific nutrients. More auxin. Auxin is a plant hormone, and less cytokinin. And less cytokinin hormone. These are all other plant hormone. And these ratios are 1:15 ratio. Sorry. अच्छा छोड़ो, वो जरूरत नहीं है, रेशियो से नहीं जरूरत. 15:1, sorry, but not required. Okay, leave it. Okay, more auxin, less cytokinin. In this chemical medium, if you put the callus, what happened? Callus start to give. Okay, rooting start. So root start to form from the callus. Suppose this is the callus. So rooting start. Further, you do one thing. What after it? You put into more cytokinin conditions, more cytokinin and less auxin. Just chemical medium you change. Where the auxin was earlier more and cytokinin was less, change. Cytokinin more and the less auxin. What will come? Now from this, where the rooting was there, now shooting also starts. Okay, what happened? More cytokine, less in this case. What happened? Shooting start. Matlab stem formation also start. Okay, and it's what? It's a entire mini plants. Ab here what you do? You just cut the various segments. Just एक part I just to cut it. One more segment I just to cut it. So 1, 2, 3, 4. Small plants you have developed into the laboratory. Okay, laboratory, everything. And now this plant you take and grow in some desired place, second wala in some other place, third wala other place, fourth wala other place, and multiple copy of this plant you can develop. Okay, but up to here, all the entire work was in the laboratory. Later on you grow in the field trial, and now the plants will grow. Tell me, plant wala tissue culture you understood or not? Plant wala tissue culture you people understood or not? I just to simplify the things that was important. But everything we are doing it, aseptic conditions. A lot of safeties and mechanisms are very, very important. Okay, it's a method of the tissue culture in the plant, guys. How even better again I'll tell you. Kindly make this picture. Okay, kindly make this diagram. Now some small topic left. First of all, we'll go into the gene therapy. Okay, so first we'll go into the gene therapy. Arey bacha, MS medium is the name of a chemical medium. Marasuz and Sku is the scientist who develop and prepare this media. Okay, not required. Only remember some nutrient medias are using. Sugarcane chip bud wala question. You just show me Stuti. Later I'll discuss. Okay. Last year I know around 7-8 questions UPSC has from the agriculture. Last year it happened first time. Ab sugar chip wala, what was the question? Whenever you see, later on I will think over it. Shamu. Micropropagation terms you can use generally for tissue culture. So generally for everywhere this term will come. Either you are going for plants or you are going for animals. Generally micropropagation term mostly people using for plants. But generally tissue culture is a micropropagation, not for only plants. Okay. So Mishi Morashewski's medium is that it's a Newton media. Its name is that, nothing more. If you don't like to, you left MS medium, not required. You left it. Okay, I just given the lot of media, MS medium, Sujex medium, Noof mediums, not required. Okay, somebody want that I just to again revise the entire thing. Yeah, okay, very simple. See, I just said, guys, I have taken any of the plants, any parts, that is called explant. Thoda sa segments of the leaf, yeah, you take it, it's an explant. And you just put this explant into a Newton media. Newton media is a food. This explant have a cells, a tissues. This cell and tissues start to grow, and the cell that was small, it will grow and become as a big size. That is called callus. Right now have a no root and no stem. Right now have a no root and no stem. This structure in the tissue culture is called as a callus. So now the cell grow and become as a callus. Callus is what? It's a mass of tissues or cells where have a no root and the no stem. So here I can tell you it's an undifferentiated mass of cells or tissues. Okay, when the cell will grow. Okay, earlier the size was very less, now the size even grow. Now some plant hormone we will use. Plant hormone medium I will develop where the auxin concentration will be high and cytokinin concentration will be low. These are the name of some plant hormones. In this plant hormone combination, what happened? You know what happened? The root start to come from the callus. Callus, there was a no tissues, nothing root and a stem. Okay, undifferentiated, it start to differentiate, become as a root. Later on the combination change, more cytokinin, the less auxin form. Now the stem formation also start, and finally a complete plantlet comes. In this plantlet have a different segments you can cut, and all 1, 2, 3, 4 you can develop as different places, and finally it will grow and become as an adult plant. It's a procedure. Okay, combination you change. It is a part of research one. What about the overall conclusions? That is important to you. Oh, sir, I understood. This is a conclusion that through the help of artificial conditions, some Newton media, okay, it's a research wala part, scientist wala part, people can develop the plants also in the laboratory. As through the help of stem cell, we can develop an organ, yeah, organisms. Okay, similarly here in the case of plants, we can also do. Okay, now come next, gene therapy. Try to finish the biotechnology, okay, before break. So after break, I'll start the AI, robotics and nanotechnology. So today and tomorrow first half, okay, that nanotechnology will over. Okay, and then we'll start one defense technology, as somebody told me. So defense technology was a discussion I'll do, and then health and disease and miscellaneous. Okay, welcome. So now come gene therapy. And then gene doping. Gene therapy and gene doping. Gene therapy and gene doping. So gene therapy is what? We are going to treat again genetic defect. Gene therapy. Therapy means therapy, therapeutic uses, treatments. So here a person, those are suffering with some genetic diseases. Those treatment we are doing. The treatment of defected genes. Treatment of defective genes. So treatment of defective genes. So what is a gene therapy? Treatment of defective genes. So treatment of defective genes here in this case. So treatment of defective genes. And genes, those are coming generation to generation. That all the basic discussion I have done. I am directly coming into the points. There are two ways through that we people can do gene therapy. There are two ways through that we can do the gene therapy. Okay, what is it? Germline gene therapy and somatic gene therapy. And somatic gene therapy. Germline gene therapy and somatic gene therapy. So there are two types. The one is a germline gene therapy and somatic gene therapy. Germline gene therapy, what happened? Okay, here therapy we are doing in the germ cells. What are the germ cells? You know very well. During the meiosis, mitosis division I discussed with you. Germ cell, generative cell or reproductive cell. So germ cell means sperm and egg. Germ cell is what? Is sperm and egg. So what treatment you are doing here in the case of sperm and egg? So the treatment you people are doing in the case of sperm and egg, it's a germ cells. Okay, it's a germ cell. We are doing the treatment inside the sperm and egg. In the case of sperm and egg, germline gene therapy. Okay, germline gene therapy. And one thing more here in this case. Okay, this is the modifications. Okay, this is the modification of genes here at this level. Here the modification of genes at this level. Here modification of genes. If modification of genes we are doing into the reproductive germ cells like sperm and egg. Okay, this will be the part of germline gene therapy. And somatic gene therapy, this one is performed in the somatic cell. This one performed in the somatic cell. Any of the somatic cell we are doing some genetic treatments, yeah, modification of genes. So if modification of gene we are doing into the sperm and egg level, that's what? It's a germline gene therapy. If modification of gene we are doing to the somatic cell, you people know very well where is a somatic and where is a reproductive cell. Now come one more thing guys, another point. Okay, here in this case, germline therapy, sperm and egg, what about the changes we will do? In this entire organism, the same genetic changes will come. And when the entire organism have a same genetic character come, so this is what? Generation to generation, this therapy will go. So generation to generation, this therapy will go. So generation to generation, this therapy will follow, and the generation is totally being correct from the genetic defects. One point. But somatic cell only to the certain part of the body. Somatic cell only certain part of the body. Only certain part of the body. Okay, only certain part of the body. Somatic cell treatment, germline therapy. Okay, it will not go from generation to generation. So not go from generation to generation. So it not go from generation to generation. So it is not going from generation to generation, you must be clear. Only certain part of the body, it is not going from generation to generation, you people clear. Only in the case of you people can go for this. Okay, so what is the germline gene therapy? Genetic modification in the reproductive cell like sperm and egg. If I have any sperm, just like very simple guys. Suppose father is a sickle cell anemia, mother is a sickle cell anemia. Both have a sickle cell anemia. Okay, so sickle cell anemia genes are here. Definitely sickle cell anemia genes are here in the both two. What you will do? You screen the sperms. And where is a sickle cell anemia? Remove it. Modify the gene, insert the normal genes. Okay, fine. Same thing you do from the egg cell. Egg cell say again you remove the sickle cell anemia, integrate the normal cells. And now the sperm and egg you fuse outside the mother body, and now the zygote is free from the sickle cell anemia. Of this zygote made baby, if it will develop, the entire cell have a what? No genetic defect of sickle cell anemia. And when this baby, this person will become as an adult, and now this person is going to give their own baby, already genes are not there anywhere inside the cell and tissues of their own body. So the generation to generation treat. And somatic cell only certain parts of the body cells. Ab just like guys, you people have a leukemia disease happen. Some genetic abnormality happen inside the bone marrow cells. Listen me. Some genetic abnormality happen in your bone marrow cells. I integrate some carrier or vector inside your body, and that carrier or vector enter inside your bone marrow cells. And what about the defect happen, the genetic defect? That's why the stem cell, that's why the WBC number increases. It will stop the formation of WBC, and now you people treat. Okay, it does not mean the generation to generation that characters will. No. Somatic cell only in the case of you. You know what happened? Cancer is a mutation problem. That is a theory based. Try to understand. Cancer is a mutation, sudden genetic changes. And I'm telling you, suppose you have suffering with the blood cancer. Those treatment I have done in the case of a stem cell. Now I'm telling you, you have a blood cancer. Okay, and your bone marrow cell got mutated. And that's why WBC number abruptly increases. What you will do here in this case? In this case, there is the best thing is what? You just enter some of the virus inside your body. Some vector or carrier you through the help of some gene you enter inside your body, and that gene will go inside your body and enter into what? Into your bone marrow cells. And now bone marrow cell start to form the normal WBC cell, and now it is what? It's a somatic cell treatment happen. It's a somatic cell therapy. Tell me everybody, you people clear or not? But here generation to generation this treatment will not go. Only in the case of you this therapy become possible. You people tell me, you people clear here or not? I want everyone answer yes sir. Just tell me guys. You people clear here or not? Germ cell and and somatic cell. Great. One more thing guys. Parallelly one more discussion I will do. The one is called chemical doping. Chemical doping. Actually, you know what the people are doing? What about the chemical doping? See guys. Athletes try to enhance their own physical stamina. For that purposes, they are taking some chemicals. That chemical name, I don't like to use it. Okay. But you know, whenever the athletes will take a chemicals, any vaccines, any medicines, so that sample will come in their own bloods, in their own urines, and then in the doping test they will identify as a positive and they fire from it. So in the case of chemical doping, persons easily find out. Many of the chemicals, Procrit alpha, a lot of chemicals people are using in the case of chemical doping. Even female athletes are taking the male wala hormones, you know, reproductive hormone, testosterone injections, because male sex hormone is very strong hormones. Okay, so that testosterone injection females are taking to enhance their own stamina. Okay, to enhance their own stamina. So this is the point I'm telling you. Okay, so chemical doping, my person will easily identify through the blood sample and urine sample. But what happened in the case of gene doping? Gene doping is what? Actually, gene doping is a misuse of gene therapy. Gene doping is a kind of misuse of gene therapy. Okay, and which type of gene therapy you are doing? That misuse of gene therapy you are doing, so the answer is a somatic wala. This somatic gene therapy misuse, athletes will do inside your body and enhance some genetic response inside your body. And enhance some genetic response inside your body as a gene doping. And enhance some genetic response inside your body as a gene doping. And in the case of gene doping, guys, some of the body organs, stamina they have increased through the help of some changes into the somatic parts of your body. Just like you people enhance your lungs alveoli, where the air entered. Normal human being this much of alveoli open, but you people have this much of alveoli open. The more oxygen start to intake, more burning of food, more energy generation. You will have a more energetic and you have a more stamina. Suppose RBC number in everyone around 5 millions. And you have done some somatic gene therapy and gene doping, and now through the help of it, what you have done, the RBC formation increase at 8 millions, 3 million more RBC, more hemoglobin, more blood fastest circulations. And what happened? Again, your stamina has increased. So in this way, guys, the people are also doing the misuse of gene therapy. But at which level have a misuse? Somatic cell. The somatic gene therapy misuse even athletes are doing to enhance their own genetic stamina. To enhance their own body stamina, and that part of the gene therapy is the misuse of gene therapy called as a gene doping. Shubham, you are right. Prithviraj, how it can be possible? Through the help of vector and carriers, because lot of the vectors and carriers are identified. Just like a pneumococci bacteria. Pneumococci bacteria where enter inside your body? Lungs. If I want to integrate some genetic character into the lung cells, so what I will take? I will take some of the bacteria that will go inside your body and affect your lung cells. In this bacteria, I just to integrate certain genes. And this bacteria when enter inside your body, it will go and inject inside your lung cells. Inside your lung cell, it enter in their own genetic characters. In that way, the genetic character will transfer to the lung cells only with the help of this bacteria. So those vector and carriers we are using. Okay, just like guys inside your class, different teachers are coming. Here I am telling you these all the different teachers are your different vectors or carriers. They are just I came here and I am entering inside your brain only science and technology part. Similarly, other teacher will come here enter only the economy part, social issue part, disaster part. I hope you understood. Similarly, a lot of carrier and vectors are developed. It is a part of biotechnology experiment. Why you think so more? Prithviraj, you clear bacha? Tell me. Prithviraj, tell me other guys, you people are getting me or not? The misuse of this somatic gene therapy is a part of gene doping. But one of the difficulty in the case of gene doping guys, athletes not easily detect. Athletes cannot be easily detect, and this is the main problem here. Athletes cannot be easily detect. Okay, because there is a no blood sample, there's no urine sample. So how you will identify the athletes? This is the difficulty here. Okay, and now it is the issue in front of WADA. So WADA, in front of WADA have a issue. WADA. What is WADA? World Anti-Doping Agency. World Anti-Doping Agency. So World Anti-Doping Agency. WADA, World Anti-Doping Agency. World Anti-Doping Agency. Biotech is very lengthy topic. 3 days it is continue, but still not finished. Today I want to finish it. Okay, I'm good. I'm so happy that everybody respond me properly. Great. And you people even connect with me also. When I ask the question, you literally respond me. And I always request you guys, if I'm taking your live classes, so why you are going to listen the same entire classes in the recording form? The reason, you know, when at a stretch you will sit here in the 4 hours, you will listen the science attack with full concentration. In the free time when you will see the recording video, so you will stop the video after 1 hour, you will go and see some dunya. You waste your time. So better live classes is here, so you should listen the live classes. So I hope you people very clear with the gene therapy and gene doping and all. Okay, gene doping and all. Now we'll come into the other one directly. Okay, so for WADA, gene doping always have a issue. Okay, and let us see. Now we come into the gene editing. Gene editing. Editing. Prabhat, absolutely, it's a very good question. Is any side effect of the gene doping? You tell me, who will say that I have done gene doping 10 years back and that's why I got a gold medal? But definitely scientists are believing it that gene doping will definitely create some problem. Definitely it will create some problem. For the short time it is good, but long time it is not safe. Just to win a match, game, it's fine. But definitely, but how to identify? If you are ready to do gene doping, scientists will do something, and then after 20 years, what is the reaction is going to be happen? So I hope you understood the reasons. Akshay, is germline gene therapy and somatic gene therapy part of RNA technology, RNAi technology? Bacha, why you people fix everything are connecting with the RNAi directly? अब दुनिया में हर चीज सिर्फ RNAi से ही होती है. Why you people? Because I have seen many of the queries, even student ask me, where हर चीजों में बोल रहे RNAi कर दे, जीन थेरेपी हो जाएगी. हाँ हो जाएगी. I ready. साइलेंट जीन में था ना, द डिफेक्टेड जीन यू कैन डू इट. एग्रीड. बट इसका मतलब ये नहीं है कि ओनली RNAi इज ओनली द रिजल्ट्स, ओनली द वे. बैकग्राउंड साउंड स्टार्ट. Better you people keep your voice na moderate. It's fine. When very high pitch you will go, the small voice is possible. I can listen the voice. It's fine. Okay, now let me start with the gene editing and CRISPR technology. Gene editing and CRISPR technology. So gene editing and CRISPR, CRISPR. CRISPR. In very simple way I will define you. CRISPR technology is very interesting topic. Very literally I love this topic so much. CRISPR technology. Okay. Actually, gene editing. What is the editing? What is the editing? Editing means some errors we are going to check. Editing means what? Some error we are going to check. Editing is not whole entire set of gene we are going to change. Some specific editing. Editing means only some specific points we are doing some corrections. Of course, through the help of gene editing, you are getting some result of gene therapy. But gene therapy is a broad perspective where gene editing is one of the part. This is the reason, first of all, I told you gene therapy. Now I am coming into the gene editing. Now come guys. Actually, guys, what happened? Earlier, when anybody have any genetic defects. Earlier, when anyone have some genetic defects. Okay. And suppose some mutated disease happen. Cancer vencer type. Suppose I am taking example. Okay. Now you people are doing the proper gene therapy wala procedure. Okay. So it was a time consuming process. And still not 100% effective. Now suppose the person is suffering with the cancer and you are developing all the gene therapies and all in 2 to 3 years. Tab to the person already die na. And still if you are adopting this technology, gene therapy wala, that traditional I told you, okay, somatic wala, somatic gene therapy and all, so still not 100% effective. And this was the reason guys, just like plasma therapy not 100% effective, similarly that was not 100% effective, and that was a problem guys, that was a problem in this treatments and all. So scientists was working from the long years that is it possible to have any kind of technology that through the help of that technology we can do the all entire process very fast and 100% effective. So what you got result here, this technology will provide you the gene editing process very fast and with 100% accuracy. This is the background of CRISPR technology. Okay, this is actually CRISPR technology. And this technology will do this role. Now here work start. Okay, so what about the CRISPR technology? I'll explain everything. Okay, to name I will write here. So first tell me guys, what was the requirement of CRISPR technology? How CRISPR technology better than the normal gene therapy? Those I just finished here. Gene therapy, that traditional gene therapy methods I told you, it was very lengthy process, time consuming process, not 100% effective process. And that is the reason in another way some new technology was developed, invented, scientists has done it, achieved it. That was a CRISPR technology. Those was fastest and around 100% accuracy. Okay, fastest and 100% accuracy. The first point, I hope you people very clear. Now what will come in the, again I'm telling you guys, CRISPR-Cas9 has a big topic. Pura how the DNA is cutting, how the things happen. I'm just coming and describing you everything in the very short way. In the 2 minutes I'll describe you CRISPR-Cas9. But first of all, you must be clear some points. Okay, and here I will write. So I hope you understood how CRISPR technology is very advanced. Fastest method of gene editing, with 100% accuracy. Of this type of cancer vencer type of problems, I'm just telling you guys, now it can be easily treated. Okay, now it can be easily treated. See, CRISPR-Cas9. This is the complete name I'm writing here. CRISPR-Cas9. What is the CRISPR? Clustered regularly interspaced. clustered regularly interspaced short palindromic repeats. Short palindromic repeat. So clustered regularly short palindromic repeat. So clustered regularly interspaced short palindromic repeats. Okay, CRISPR-Cas. And Cas is what? Cas is what? Cas here C represent as a CRISPR. First of all, the terms you must be clear. Here C represent as a CRISPR, AS represent as a associated, and 9 represent as a protein 9. So what is the full form of Cas9? CRISPR associated protein 9. It's a very specific protein. CRISPR associated protein 9. CRISPR associated protein 9. Okay, CRISPR associated protein 9. Actually, you know what happened? Suppose some genetic defect happen inside your DNA. You people know very well what is the DNA. Okay, inside your DNA, some genetic defect happen. Some genetic defect happen. So some genetic defect happen inside the DNA. Some genetic defect happen inside your DNA. And suppose this is the defected gene. Suppose this is the defected gene. Achha, you know one thing guys, some of the background I will tell you. This CRISPR-Cas9, that the complete name is the CRISPR is the cluster regulating interspaced short palindromic repeat, and the Cas is the CRISPR associated 9 protein. This was the very specific genes and proteins. Oral I can tell you as oral proteins. Those even discovered in some of the microorganisms. Yes, this mechanism was discovered in some of the microorganisms. From these microorganisms, you just take CRISPR-Cas9 separate from the organisms. Suppose some of the bacteria have a CRISPR-Cas9 proteins. Okay, you just take the CRISPR-Cas9 from this bacteria and enter inside your body. Enter inside your body. And very specific character guys, through the help of CRISPR-Cas9, wherever you have a defect inside your body cells. Okay, suppose some mutation happen, some genetic defect happen, and this is the green color wala abnormal cells, abnormal DNA sequences. This is some abnormal DNA sequences. What is it? This is some abnormal DNA sequences. This is some abnormal DNA sequences. So what here, what will come? This Cas9 will come here at this place, come here at this place and cut this DNA, and cut this DNA, and cut this DNA, and finally what happened? This portion is totally replaced outside. And see, entire mechanism it is doing very, very fast. And what about this? Cluster regularly interspaced short palindromic repeats. Okay, even those sequences here it is going to cut. Those sequences it is going to cut and remove outside. Okay, it's abnormal sequences. Those was made here because of mutation and something. Okay, now here what happened? This palindromic sequences. Palindromic sequences are what? Front and the back, both sides have a same sequences order. Just like suppose Madam I am not Adam. Madam I am not Adam. I am not Adam. ADAM. Okay, yeah, Madam I am Adam. The better is just a minute. Madam I am Adam. You just see. Madam I am Adam. This type of sequences are palindromic. Left to right, right to left you go, you will potentiate the same. Okay, see, Madam I am Adam. Left to right and right to left, you know, sequences are same. In this way guys, those defected gene are going to remove outside by the help of Cas9. Cas9 here work as a Caesar. Caesar, you know, Caesar, to cut the sequence. So defected gene directly it will go inside the body and remove it. and the normal sequences, CRISPR palindromic sequences are going to fulfill. So ultimately those changes happen, further it will correct and the sequences are right. Sequences are right. Just write 2 line here. Okay, just write 2 line here. And the normal sequences, CRISPR palindromic sequences are going to. Okay, now come here. Right, how it works, right? How it works. Tell me guys, I just to simplify the concept of CRISPR-Cas9 here or not. First tell me yes. Oh, without asking me the people respond me Najasha, good, plus, good. But again I am telling you very fastest recovery. And this type of CRISPR-Cas9 was identified in some bacteria. And how a bacteria are become important to you guys. Okay, even soil bacteria you have seen for the formation of BT crops. You have to recall. Okay, now write about the how it works. Right, how it works. This technology works like right. In 2 lines you just write it. Don't go more technical, right? This technology works like a cut and paste mechanism. This technology works like cut and paste mechanism on DNA strand that contain genetic information. Okay, first off, the DNA strand, strand, right? The DNA strand, strand, that DNA strand identifies the specific location of a gene, specific location of a gene that need to be changed, are edited, or are edited, edited, are edited, change are edited, and then using Cas9, protein, and then using the Cas9 protein, which acts like ACTS, acts like a Caesar, SCISSOR, which acts like a Caesar, and cut off the defected gene, First stop. Once the DNA strand is broken, it has natural tendency to repair itself. It has natural tendency to repair itself. First stop. During the auto repair process, scientists will supply the desired sequences of gene, supply desired sequences of gene that bind itself that bind itself that bind itself that binds itself with the broken DNA strand, bind itself with the broken DNA strand, itself with the broken DNA strand. So it's a CRISPR-Cas9 and have a lot of hope. 2 times UPSC asked the questions at one I think in 2020. What is CRISPR-Cas9? So it was a faster gene editing process. UPSC asked in 2020 just a very simple questions. I think I have shown you that question also. Okay, because it's a question paper, so I can show you. This was a very simple question. 2 times UPSC asked the questions. See the one question that 2020 you can see here right now. Okay, you will know these questions like pro-nuclear transfer is used by 2020, prevention of mitochondrial genes in the next offsprings, the one question. Another question that UPSC has, biotech I told you guys at least every year 2 questions so UPSC is giving you. Okay, the one I think it was a CRISPR-Cas9 also there. I think in 19 it was there, not in 20. Achha, you tell me guys, right now you people can think over it or not, because you have a lot of information about the gene therapy and all. Can you tell me what will be the correct answer in the 4th questions? Because it was a last year question paper. I just want that you people can understand. Even this answer is bold. I think you can find out the answer very easily. But I just want that you people okay, you just tell me you people can understand these questions or not. And I just want that you people understood the answer is a D 1, 2, 3 or not. Reason a lot of discussion I have done here. Okay, and I just want to show you guys like what about the effort I have given here. Okay, that you must be clear. And I hope everybody have this PDF. Everybody have this PDF. Because institutions has given you, all of you, this PDF. You will tell me this answer, you people are getting clear that why the D answer is right. It's a 2020 question papers. Biotechnology last time have a 3 question in your paper and all 3 questions you can solve. All 3 question paper you people can solve. Consider the following statement: genetic changes can be introduced in the cells that produce the eggs or sperms of the prospective parents. So genetic changes we can do under the cell that can produce the egg or sperm product. Yes, that is called germline gene therapy. I hope everybody clear the first one. And somatic cell we can also do. See, question paper are very easy. The problem is now the few people think so the science and tech is a current affairs. Damn idiot guys. Okay, but I cannot say the problem is you people are misguided. But it's a current affairs. Tell me guys, it's a current affairs. Is it current affairs? It's a static part. So every questions are coming. 80% plus questions are coming from static parts. This is the reason I said that you people share to everyone, and I said to all of you connect with me on Telegram. Even any of you people have a friends, and literally you want that your friend should also clear the UPSC. So you just advise and share my Telegram to connect it. I just want to connect all the people, those are serious aspirants, and those people have a myth with the science and tech actually. I want to resolve that problems. Second one, a person genome can be edited before birth at the early embryonic stage. अब you tell me guys, the person genome can be edited before birth at the early embryonic stage? Definitely. In the embryonic stage, we can also do the treatments and all. Okay, that will be the part of somatic cells and all. A person genome can be edited, and here how we can do the addition and all. Okay, person genome can be edited before the birth at the early embryonic stages. Third, human induced the pluripotent stem cell can be injected into the embryo of a pig. हाँ क्यों नहीं कर सकते? Any of the you just tell me guys. Okay, mammals have a stem cell, you must be clear. Human induced pluripotent cells, okay, can be injected into the embryo of a pig. See, here the answer is can be injected. So can be injected, definitely, why not? Okay, can be injected, why not? Okay, so it's a general and behavior questions. One more questions, those UPSC asked. Okay, UPSC asked. Okay, those UPSC asked. It was a biotechnology wala question, I think 2019, not 20. Let me check. RNA interference wala question you people are able to solve. See. With reference to the recent development in the science, which of the following statement is not correct? Functional chromosome can be created by joining the segment of DNA taken from the cell of different species. Okay, you just tell me your chromosome sequences you can develop with the help of taking of DNA from other sequence. No, other species. Same species based CNS sequences you will take it. Because you people have a 3 billion base pair of human have a same. So if you will take another gene of pig or any other one, so that will be the another genome, na? Oh, so of course the first statement is not correct. Same species member you will use it in that cases. How other species member you can use it? RNA interferences you people know very well. That discussion had, yeah, that was the question. Sorry, it was not 20. Extremely sorry, it was in 2019. Tell me guys, what will be the correct answer here? Okay, everyone. So yes, you people are right, absolutely right. So I just telling you guys the question paper is very superficial type. Don't worry, whatever the discussion I have done now, it will be enough for you. Okay, and don't be misguided by the science and technology. Here still you people can solve 80% question very easily. Okay, there is just a highlight. And now I'll come into the last about some terminology. And then biotechnology will over. Some terminology. Okay, some terminology. And this all the terminology I will finish in 30-40 minutes. Some terminology. So lot of the discussion I have done here in this case. Okay, gene editing and all, everything is over. Now some terminology you will come and just you start with the biosensors. Just 2 minutes I will take with all biosensors. Okay, biosensors. The one discussion is the biosensors. Second discussion. Okay, I'll write the topics and then I'll finish as soon as possible. Biochip. Biochip. The second one is a biochip. Okay, second one is a biochip. Third one is a biosignature. But biosignature I have discussed somewhere. I know very well you will respond me during the discussion of biosignature. Biosignature. Next one is biogenerics. Next one is biogenerics. Okay, next one is biogenerics. Next one is bioplastic. So next one is a bioplastic. 6-7 topics only here. The next one is a bioplastic. Okay. Next one, biological weapons. Biological weapons. And the effect of biological weapon is called as a bioterrorism. This is called as a bioterrorism. Actually, CRISPR-Cas9, who asked me, Shubham, have a lot of issues also. Nahin. What is the logic CRISPR ban? Actually, you know, CRISPR is not completely banned. Many people are doing the work because of ethical issues. It's not being there, guys, that it only do the correction of the sequences. It may also create some abnormalities into the living organisms. Okay, because fastest of seizures and all, and if abnormality happen, तो definitely it will create a massive problems, because of lot of the genes have an interaction inside your body. Okay, lot of the gene have an interaction inside your body. Okay, if any single gene it was going to change, so other interaction can also be disturbed. Okay, because the means is not natural, the means is through artificial ways we are going to proceed. Okay, we are going to take artificial means and some other reasons also, those are not required, so I just skip it. But still the people are doing the work on the CRISPR-Cas9, and even recently China has done, India is also working a lot on the CRISPR-Cas9 technology. So CRISPR-Cas9 is working, don't worry. Issues are here, so somewhere have a ban and something here. Okay, वो तो हमेशा होता है, it's not done new, it's happen everywhere. Okay, it's happen everywhere. And biosparging, and biosparging. So these are the some few topics here. Biosparging. So these are some few topics here. Biosparging. CRISPR can be possible in both somatic and germ cells. Generally, CRISPR we are doing in the case of somatic cells. Okay, what about the report I got it? It is in the somatic cell. Okay, so what about the report I got it? It is in the somatic cell. Okay, reproductive में I never heard it. Okay, I could not get anywhere has an article कि germ cell में we cannot do the CRISPR. But in my knowledge, those I read the article, everywhere the answer was a somatic cell. If anywhere have a germ cell, तो मुझे दिखाना. I will also go on Google वाले बार देखूंगा, but I think so it is a somatic cell generally. Okay, somatic cell generally. Okay, but possibility definitely on the germ cell. But what about the results and everything happen? Here the people have done the research and everything on the somatic cell. Okay, but it's a good question. I will also see. It's literally Akshay. It's only the somatic cell or germ cell. Germ cell I will see. Not very sure, but I'll see. Okay, now come here. Biosensors. Biosensors also called as a biological. I think I take a break right now. Okay, and then after break I will continue. Around 20-30 minutes this whole the topics will take, and then next I'll start the AI, robotics, and nanotechnology. So AI, robotics, and nanotechnology. These chapters are boring, I should not say, but factual I will definitely say. Fact. But see guys, facts one time it's important. Last year UPSC has one question from AI, I think last year, and one more question UPSC has from nanotechnology. I hope you people know very well. In 2020, those questions UPSC has from nanotechnology, it was a CNT based. Nanotechnology is the most important. AI and robotics not. Okay, but AI was the essay question in 2019 essay. One of the questions. Most of the people have written the essay from my book. Because AI is even not given in your syllabus. Your syllabus have mentioned for the robotics and nanotechnology. Okay, mostly these topics are for your mains paper, but nanotechnology somehow UPSC also has in your prelims paper. CNT was UPSC questions. Graphene was UPSC questions. I'm telling in the past 10 years. And one more wala drug therapy and drug targeting wala questions, that nanotechnology can be used for drug therapy, drug targeting and all. Okay, drug targeting, gene therapy. Okay, gene therapy and all through the help of nanotechnology is possible or not. Okay, so these were the three questions the last 10 years, I think 8-9 years UPSC asked from nanotechnology. So just after it I'll start it. But again I'm telling you, this is entirely fact and figures. A lot of fact and figures I'll tell you, so parallelly you people will write it. Yeah, if you have a book, so book ko kholna aur underline karna chalna. Okay, those have not the book, I'll write on the boards. I'll give you proper time to finish it. Iske baad, tomorrow I will start defense. Again I'm telling you, tomorrow I'll start defense. Okay, so tomorrow I'll start with the defense, and then I think not much topic. Miscellaneous topic I'll start miscellaneous topic, and then the last health and disease. It's over. So tomorrow I think defense and all finished, and miscellaneous topic day after tomorrow I'll start. Friday, I think so, maximum Friday. Biofruitigations is also come under science and हाँ कर सकते हैं biofruitigations. Health वाले issues में biofruitification and all we can do. So okay guys, meet you after 15 minutes. Okay, it's a break. And now the topics are not more technical. It's a information based. Okay, it's a information based. Hello. I think I'm audible to everyone. Your question I'll take later on. Okay, first you just finished me. Okay, but it's an entire geography. It's an entire sugar cane. See here, some of the part is actually अच्छा बताता हूँ. Just wait. Okay. Your question I'll take later on. Okay, first you just finished me. Some terminology part because still biotechnology is not going to over. So first of all, I just want to finish it as soon as possible. What is a biosensor? Biosensor is a biological sensor. Okay, biological sensor. And better guys, I'll give you directly examples. And through example, you will get it very easily what is a biological sensor. Even Prega News for the detection of pregnancy through the help of urine test. Even insulin means sugar level, sorry, not insulin, it's a sugar level inside your blood, sugar level inside your blood. These are very common examples those you people can see in your surroundings. So this type of identification we are doing, like pregnancy test, sugar cane test, that we are doing it here in this case. Okay, those instruments we are using, it's a kind of biological sensor. Okay, so I hope it's very easy and you can understand what actually the biological sensor is. These all devices are whatever the system is available, you are purchasing from the market, pregnancy and sugar content, it's a kind of biosensor. Okay, and what is the function of it? They are using the biological components. They are using the biological components. The biological components may be your blood, maybe your urine. Okay, and through the help of physicochemical detectors. Okay, it's a kind of detectors. Those are physiochemical detectors. Okay, so biosensors are called as a physiochemical detector. Those are using the biological components for the detection of various activities inside your body, various feature inside your bodies. Okay, it's a sensitive biological element is used there. Maybe anything can be possible there, guys. Okay, the best biosensors are what? Definitely they should have a small size. Okay, they should have a small size. They should have a small size. Fastest results, you know, these are the better. Small size, fastest result, and not costly. You know, they are the best. These are the best. Biological sensors. They should be not very costly. Small size, user friendly in this way, guys. Even a lot of biosensors even also develop. Those biosensors can detect about the pollution level. Okay, even they can use the biological weapons also. Means a kind of a harmful bacteria and viruses from the atmosphere. Okay, in that way biological sensors even also has been developed. A lot of the experiments we are going to do. Okay, especially for the pollution, to check the pollution and biological sensors. Okay, biological weapons and all. So in this way biological sensors are very, very important. Even for the detection of so in the case of war vagera, military purposes, maybe biological weapon, chemical weapons, you know, that all the detections also become possible through the help of various sensors. Okay, so it's a future, nothing more. It's very simple concepts. And biosensor were once UPSC question in the mains paper around 2008-2009. Okay, but you people must be remember it was a question in mains paper of biosensors around 2008-2009. But still, you know, it's very common topic. So you remember biosensors. Okay, now come next, that is called biochip. Okay, next come that is called biochip. Okay, biochip. Actually, biochip has two components. biochip have two components. Directly I'm coming into the structural parts, two components. Okay, the one those are going to install into the living organisms is called as a transponder. This is called as a transponder. And second, those transponder is implanted inside your body. Through the help of reader, we are going to detect it. Through the help of reader, we are going to detect it. So one is the transponder and second is a reader. Corona testing kits are also being a sensor. I agreed. Okay, some of the sensors we are using it. Okay, but does not mean कि PCR को भी आप sensor बोलोगे. नहीं. Some sensors even develop for the detection of COVID. Okay. Okay, some sensors people have developed for the detection of COVID. So transponders and readers. Actually, transponders are those electronic device we are going to implant into the living body. Implant into living body. Implant into living body. Our readers are those we are sending the signals, and the signal is the radio frequency. Signal is what? In the form of radio waves, and radio is a non-directional, so it's go everywhere. It's go everywhere, and where inside the organisms, your living organism body have this transponders. Okay, they will do the detection of it. And transponder always in the passive mode. Transponder always in the passive mode, so they don't have any need of any batteries or anything. They don't have a need of any batteries. Just like this pen is in passive mode. पता है इस डिजिटल पेन में कोई भी बैटरी नहीं लगती, ये पैसिव मोड है और ये हमेशा it will work. So similarly guys, this transponder is what? It's in passive mode. Okay, it is in the passive mode. Passive mode means no extra energy is required. Neither you tell me, suppose in the terrorist body, okay, you implant some small chip as a transponder. So after 2 years battery finish and they call you, bhaiya, you come, I want to change your battery inside your transponders. Neither transponder will not work. No. So it's in passive mode. Okay, it's in passive mode, and through the help of radio frequencies we can detect it. For roaring animals, cattle, they are moving somewhere, you can use it. Okay, for the safety and securities of animals, for the detection of terrorists, you can use it. Okay, in that way that can be used. Even not only this guys, some other purpose of biochip, even in the case of health monitoring. Health monitoring we can also do. So we can also do the health monitoring. We can do a health monitoring of a patient. So health monitoring of a patient can also be possible through the help of biochip. Okay, through the help of biochip. Okay, health monitoring of patients. Suppose guys, some heartbeats. Okay, somewhere have a problem. Okay, so inside the heart you can implant the chip guys. I can tell you it's a part of IoT. Somehow you can connect with the IoT also in Internet of Things. I hope you remember IoT at that time I discussed with you. Okay, the small chip will do the monitoring of heartbeat rate, and the doctor mobile phone will connect. Every moment after, a doctor can find out all the activities of the human beings. Okay, so properly a heart monitoring we can do through the help of it. Okay, properly a heart monitoring we can do through the help of it. Okay, properly heart monitoring we can do here through the help of it. Okay, so properly heart monitoring we can do here. Okay, so it's a biochip. I think you can understand, nothing more here in this case. Okay, you people remember it. Even in the coming future guys. Okay, in the coming future, this is what? As electronic device. It is an electronic device. This electronic device we people can also use for the coming future. Okay, for all the data safety. Just like guys, suppose in the coming future a small chip implant inside your body somewhere, and no need of your wallet, not required, your ATM, your everything, your all the informations inside the chip, it will be safe. So just a police personnel, yeah, ATM, or wherever you go, just scan your chips and all. That is the part of what? Biochip in the coming future. Your every information you can save inside your body. You no need to carry extra wallet and all. Okay, so in that way biochip may have a good future also. So let us see what about the possibilities in the coming future with the help of biochip people can do. Okay, so your ID card, insurance, every information you can connect. Okay, so finally it is a small chip, those are going to implant inside your body, and the chip is called as a transponders. Okay, fine. Next you come biosignature. Biosignature I will not discuss, reason you people know very well. You tell me where I discuss with you about the biosignature. Let me see. You people heard it, biosignature. Where you people have listened me in the case of biosignature? Manjushi, it create a radiation effect and all. Inside our body, we are safe. We are 100% every time we are under the effect of radiations. Here inside the room, those classes I am taking, Wi-Fi is there inside my room. Tower is in nearby. So you people are every moment of time under the effect of radiations. Do you know or not? Okay, 100% you are under the effect of radiations. Why you are thinking a chip will go there? अब ये tell me एक बंदे का हार्ट अटैक आ गया, मर सकता है कभी भी, उसके केस में बायोचिप लगा के उसको बचा रहा हूँ, उसको मार रहा हूँ. You tell me answer. You see your priority. You see your priority. Yeah, absolutely right. In the case of space tech, I discussed with you. During the case of Chandrayaan 2, I told you that rover was some planning. Rover will take a soil sample of moon surface and do its chemical testing. And through the help of chemical testing, it will try to inform us about the presence of past life and the present life. So you people know very well, biosignature type of terms generally coming in the geology, study of earth and rocks. And through the help of geology, guys, samples, any of the planets, any of the celestial bodies, okay. So, okay, through the help of geology, earth and rock samples, we can find out the past life and the present life. We can try to find out the past life and present life. This is the overall. Chandrayaan 2, absolutely right, guys. So biosignature, you people know very well. Okay, biosignature you people know very well. One more, biogenetics. 1 minute only biogenetics I will take. Actually, application of biotechnology in medicines. Application of biotechnology in medicines. Absolutely right, everybody right, 100% right. I'm so happy that you people are following me properly. Okay, following me properly. Okay, so biogeneric is what? It is the use of biotechnology in the medicines. You know, if you people are doing the biochemistry, microbiology, biotechnology, some degree courses. Later on, they join a job in the companies. And whenever they join a job in the companies, guys, Ranbaxy, Cipla, Dr. Reddy's, this industry is a biogeneric industry. Simple. This industry is a biogeneric industry. Okay, where the living organism we are using, chemical compound studies, biotechnology we are using for the development of different medicines and all. So everything is what? Everything is a part of biogenerics. So what about the biogeneric and biogeneric industry? I hope you people understood. Okay, now come bioplastics. Bioplastic, I'll take some time. Okay, bioplastic, I'll take some time. Small topic, guys, small topics. What about the bioplastic? Okay, bioplastic also called as a biopolymer. One more term is used there. So either you call bioplastic or biopolymer. Bioplastic or biopolymer. So bioplastic or biopolymer. What is this bioplastic and biopolymer? See guys, I have a questions. What is the source of plastic? Anyone? What is the source of plastic? What is the source of plastic? From which you people develop a plastic? Because plastic not available in the nature. So from where you people develop a plastic? Anyone? What is the source of plastic? What is the source of plastic? I hope somebody will give me a satisfactory answer. So what is the source of plastic? Yeah, absolutely right. Those answer I got it. It's a crude oil. Agreed. Okay, good. It's oil. Agreed. It's a crude oil. Those crude oil you are taking from the other country, guys. From this crude oil, we are going to develop a plastic. We are developing a plastic. And this plastic is not safe. Everybody know. For the environment, for the living organism, it is not safe. Everybody know. Plastic is not safe from living organisms. It is not safe for the environment. Not safe for the soil health. Okay, soil health. So everywhere have a problem, and some other problem also there because of plastics. And some other problem also happen because of plastic. Okay, fine. Actually, you know guys, these plastics earlier was not a problem when they were not a plastic. They were available into the fossil fuels. But whenever we people have developed the plastic is a synthetic material. Plastic is what? Plastic is a synthetic material. So plastic is what? This is a synthetic material. So plastic is what? This is a synthetic material. So plastic is a synthetic material. Those develop from the crude oil. Okay, those develop from the crude oil. Next point. Okay, see, those plastic as a synthetic material developed in the crude oil. After its development, they being as a problem. And this type of substance are the part of xenobiotics. But xenobiotics term is the environment term. Xenobiotics term is the environment term. Is the environment term. What is the xenobiotics? What is the xenobiotics? Okay, you people write the xenobiotics. Okay, xenobiotics. Actually, what is the xenobiotics? Xenobiotics are those substances we are developing from the natural sources. Xenobiotics are those substances we are developing from the natural sources. So xenobiotics are those substances we are developing from the natural sources. After the formations, after these formation guys, they become as a problem for the health and environment. These all are called as a xenobiotics. So xenobiotics are those compounds are developing from the natural sources. Similarly, I can tell you example of pesticides. Example of chemical fertilizers. They are also developing from the various natural products. But when as a synthetic material from the natural product we develop the chemical pesticide and fertilizers, they still being as a problem for the environment and the health. They still being a problem for the environment and health. Okay, they still being a problem for the environment and health. This is xenobiotics. This is xenobiotics. Okay, this is xenobiotics. Okay, this is xenobiotics. So similarly, I can tell you the plastic is also what xenobiotics, but it's an environment concern. So I hope you understood the natural, even those plastics, synthetic plastic we are developing from the crude oil, it's a problem. But as a alternatives, I am giving you bioplastics. Bioplastic is not, its source is not the oil products. Its source is what? Its source is a biological products. Its source is a biological products. And the mode for the formation of plastic, it's a what? Microorganisms. It's a what? It's a microorganisms. It's a what? It's a microorganisms. And source for the formation of the plastic is what? It's a microorganisms. Actually, you know, the first bioplastic that was developed. So the first bioplastic that was developed, it was the PHB. You must be clear. The first bioplastic was developed, it was a PHB. The first bioplastic was developed, it was a PHB. PHB means polyhydroxybutyrate. It was a polyhydroxybutyrate, PHB. And with this polyhydroxybutyrate, that is the world's first bioplastic, it was developed by the help of bacteria, Bacillus megaterium. Bacillus megaterium. Sources biological products. Later on, some more bioplastic developed like PHA, like PLA. So polyhydroxy alkanate, alkanate, polylactic acid. These are few name of bioplastic I am telling you. Polylactic acid. I'll tell you what is the source of polylactic acid. See, you just go in some vegetable shop वगैरह. See, a lot of vegetables are destroyed every day. What are inside the vegetable? A potato. Potato have a starch. What is inside a potato? Potato have a starch. Okay, you do one thing guys. Potato have a starch. Those potatoes going to destroy. Okay, potato have a starch. In this potato starch, you do some microbial activity. Okay, some bacteria वगैरह we are using it, and through the help of microbial activities, this starch will convert into lactic acid. Okay, scientists have achieved it, guys. I'm not anything created here. It's already successfully happened with the help of certain microorganism, bacteria. Okay, what happened? The starch of potato that was totally destroyed in the market, guys. Okay, that's being converted into lactic acid. Many lactic acid, lactic acid, you join together. And when you people will join the many lactic acid, it will become as a polylactic acid. If many lactic acid will join together, it will become as a polylactic acid. And polylactic acid is what? Polylactic acid is a plastic. It's a what? It's a biological sources. And that's why it's called as a bioplastic. What is the benefit of this plastic? After this use, this plastic, if you bury it into the soil, guys, they can be decomposed very easily. Okay, because the sources are biological sources and they form because of microorganisms. So I hope this type of example, PHB, PHA, PLA, here you will also see the role of biotechnology, application of microorganisms. Tell me guys your answer. I think nobody have any problem in the understanding of bioplastic right now. Tell me fast. Nobody have a problem in this understanding of bioplastic right now. What is source of PHA? अच्छा PHA अब क्यों जा रहे हैं यार? I just given you one thing. Okay. वैसे भी I don't know what is the PHA, but इतना ज़रूर है, I am very sure some biological products are there. But just some of the name as a factual data, PHB, PHA, PLA, I am telling you. So I'm not very sure about PHA. Okay. आप देख लो एक बार गूगल पे. Okay. But this is just a information. I hope you understood how much this is better. How much it is better. How much it is better. Now come two more concepts, biological weapons and bioterrorism. This is the misuse of biotechnology. This is a misuse of biotechnology, and you people know very well, definitely it's a misuse of biotechnology. Okay, very simple guys. You do one thing, many genetic, like COVID, you can take a best example of COVID. Somebody's are telling that COVID type of viruses. Okay, people has done this multiplication to the laboratories like China, and the China spread the COVID everywhere. I don't know it's much correct. Okay, reason China is still suffering the COVID issues, but how much the truth I don't know guys. Okay, and massive level of problem China faced. So why China will develop to create this type of problem in their own nations? Maybe possible because lot of the dictatorship in the world. Okay, North Korea wala you can see, China wala you can see. Yeah, France wala you know very well, Putin. Okay, how much these people are dictatorships. Okay, dictatorships. Fine. Same thing you can see our honorable Prime Minister, dictatorships. For you all people, you make a rules and regulations, but for me, I'm free. I can do whatever I want. Okay, so dictatorships. People are not feeling, but I think so. It's a dictatorship. I still remember I have posted some of my Facebook, some information I posted. I was earlier to Facebook account, and just to post something about it against Narendra Modi. I was not. I'm BJP, but I'm again anti-Modi. Okay, I'm a BJP person, but I'm anti-Modi person. Okay, I'm totally telling you. But the person blocked my Facebook account. You'll not believe me. Well, right to speech is what here. There's nothing I said it. In that platform, I just said, see, Narwin and some of the prime ministers just organized some party in their own house, and the member was 100 there, more than 10. Okay, but our prime ministers in this COVID, for everyone to follow rule and regulations, but he visited into the Bengal elections. Just this was the information there. Okay, so then later on I stopped to share anything else like that. Okay, the people want to, shut my mouth forcibly. So it's dictatorship. But here right to speech is not there. The problem is it. But leave it. Okay. Leave it. Does not matter. We are in India, so better. Okay. Acha, leave it. So earlier this man I like so much, but because of this incidents and everything, whatever I'm seeing from the last 1-2 years, I understood. This man is still doing the politics and all. But leave it. Does not matter. Okay. We are in the class. Better we don't go on the politics. Okay. It's very worst. Because politics means what? Just to destroy the people's life. Better you people come and think over it. So COVID, I'm telling you, the COVID viruses people are going to develop in the laboratories, and they are misusing it as a weapons. So it is not good. And application of biological weapon is what? That is bioterrorism. Okay, application of biological weapon is what? That is a bioterrorism. Yeah, Lekha, I agreed with you. Few examples even I have given in my book also. The anthrax was there. That is very common. In 2010, as a biological weapon, anthrax was in the news. Anthrax is the bacterial disease, and the bacteria which are responsible to cause this effect, that's called Bacillus anthracis. It's a soil bacteria, guys. And this bacteria even infect not only human being, even also many cattle. Okay, so cattle and human beings, everybody will get the exposure of it. Okay, everybody get the exposure of it through open wounds. Okay, in inhalations, this problem can becomes. Okay, mode of contact is inhalations, any wounds, and it affect your pulmonary systems, your respiratory systems. Okay, so this type of misuse of biological biotechnology, many of the biological weapons. Okay, now the last, biosparaging. Now the last is a biosparaging. Biosparaging. Actually, guys, we people can also use microorganisms. Okay, we people can also use the microorganism to clean water. We people can also use microorganism to clean water. So we people can also use microorganisms to clean water. So a lot of water pollution and everything. Okay, so microorganisms used to degrade the pollutants in the soil layer above the groundwater. Okay, so to clean the water, those microbial activity we are using, this process is called as a biosparaging. Even many of my friends was working under the water purification system. When I was doing my research, so some of my friends was working. They were doing a lot of the procedures adopt. They were collecting to the sewage water and they totally properly clean it. But the cleanliness is for the irrigation purposes. The cleanliness was not up to for drinking water. This much of water not treated there. Okay, in the many of the parks, those water supply is going to there. Okay, actually, just in front of IIT Delhi, there was a rose garden, I think. Okay, rose garden was there. Okay, अब तो भूल रहा है, बहुत पहले की बात है. I was there. So here those water irrigation wala that people were using, ये sewage water वाला treatment था. But some of the people even they take it as a drinking water. वो जो sewage वाला जो पानी treat होगा, it's being as a white, totally clean, used for irrigation. It was not drinking water, guys. It was very dangerous. Drinking water has another site of the rose garden in front of IIT Delhi have it. Okay, so that type of water not for drinking water. So here I'm telling you guys, like through the help of microorganisms, we people can also treat the sewage type of water. Actually, those sewage water is there, have a lot of bacteria, lot of organic waste. A lot of things are there. So many processes are adopted, ultracentrifugations to put the biological masses, compounds, very small pieces. Okay, then provide the aeration also. Okay, aeration also, filter also. Many processes are, and through the help of microbial activity, those organic masses there into the water body, it is going to what? Destroy, decompose, and then become as a small particles. So somehow microorganisms we are also using under the water purification system. So those water have an impurity, yeah, the water is into the water table, yeah, aquifers. Okay, we people can treat through the help of microorganisms, and this procedure is called as a biosparaging, nothing more. The place replacing it. The rose garden now construction is taking place in replacing it. Okay, Anil bhai. I know very well the rose garden have a lot of things. Okay, I was there in my period of time to play badminton mostly. Okay, ab chodo wo sab, that is not. Okay, because the metro is there in this entire region, I know. So this was something about it, nothing more here. That's a miscellaneous topic, just 2 minutes. I don't like to waste more time because basically these all the topics are only for prelims, not for mains or any other. So you just remember. Now you come into another chapter. Those I was waiting for it. Okay, and that is AI. AI is somehow I will try to create the interest. AI. So artificial intelligence. Okay, artificial intelligence, and it was an essay in 2019. Okay, come here, AI. AI. Still it is not mentioned in your syllabus, but very important topic. Even those robotics example I will discuss. This is the part of AI. Robotics is what? It's an example of AI. AI is what? Artificial intelligence. Artificial intelligence. So AI is what? Artificial intelligence. Artificial intelligence. What is it? So we start from the zero level. Artificial means synthetic. Synthetic, not natural, simple. Artificial means synthetic, not natural. And intelligent terms always connect with the brain creativeness. It's a brain. Intelligent terms always connect with the brain. So artificial means synthetic and intelligence means brain. Intelligence terms, its synonyms always represent as a brain, because brain is the intelligence. Okay, so don't think so about it's a synthetic brain. But through the help of these terminology, I will define you what actually AI. Listen in the form story. Actually, entire scientist community, they were thinking about it. They think about it earlier. Is it possible that we people can develop a machine? Those machine will literally work like to the human brain. Again, I repeat. Is it possible to develop a machine those will work 100% like to human brain? So it was a thought always in the scientist's mind. Slowly, gradually, through the help of various research work, through the help of many R&Ds. Okay, they reached up to certain level. They have reached up to certain level where they are trying to develop a machines, some programmings. Okay, and finally they have achieved a characteristics that will work like to the human brain. So finally, what was the reason behind AI? That machine can work like human brain. That was a overall conclusions of these discussions. A machine can work like to the human brain. That is the behind artificial intelligence. So artificial is what? A kind of machine. And work like to the what? Intelligent human brain. Then the term brought as artificial intelligence. So why the term brought as artificial intelligence? A machine work like human brain. What about the conclusion of this discussion? A machine work like human brain. Like human brain. So here machine is what? Here machine is the artificial device and the human brain is represented as an intelligence. So I can tell you artificial intelligence. I have started this topic from 0 level. Again I am repeating. More technically I don't like to go. Okay, you start from the 0 level. Now another question. Is it possible that a machine can develop and want 100% like to the human brain? So here have some queries. Here have some discussions. Definitely a machine can work 100% like to human brain. 100% like to the human brain. This is a question mark. Okay, this is again a question mark. That literally a machine can work 100% like to human brain. Okay, now come here. That literally a machine can work 100% like to human brain or not. You just come. Actually guys, there are two types. There are two types here. Two types here. What? The one is called as a weak AI. Here on this aspect, I am telling you two types of AI. If I want to check that literally a machine can work 100% like to human brain, so here I am telling you AI are of two types on the basis of its a performance. As a three terms, I will use multiple times, and I want that everybody speak 10 times these three words. Even in the case of AI, robotics, and nanotechnology, three terms I will use again, and again. What are these three terms? Performance, accuracy, and reliability. Performance, accuracy, and reliability. Three terms you must be clear: performance, accuracy, and reliability. So performance, accuracy, and reliability, three terms are coming here. And I just want everybody tell 3 times at least 3 terms. Performance, accuracy, reliability. Tell me, very important. Performance, accuracy, reliability. Performance, accuracy, reliability. And now everybody write performance, accuracy, and reliability. 114 people are watching it. I just want to see that how many of people give me the respect. Performance, accuracy, and reliability. You write. Because these 3 terms, again I am telling you, for the next 3 hours in the AI, robotics, and technology, 100 times I will use. Even any of the questions in your paper, in the mains paper is coming, so these 3 terms, and definitely one question always come. Performance, accuracy, reliability. Okay, Raju, thank you, thank you. First of all, Raju, you just tell me those I told you. Raju, thank you, thank you. After your lecture, reading the reference book easy. Don't worry. Yes, performance, accuracy, reliability. All three terms. Great, great. Okay, great, 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 great. Many people smell it. Okay, good, good. You people are giving me respect. Okay, good, good. Actually, whatever I am doing now, it's for you. That's why I told you this performance, accuracy, and reliability term. Never forget. Ab now you come into the AI. Great, great. I'm so happy. Fine, fine. Great, great. So AI. So now come here. So three terms on the basis of performance, accuracy, and reliability. I'm telling you there are two types of AI. Okay. So this AI basis I'm telling you here the machine literally work like to the human brain in the respect of performance, accuracy, and reliability. Now two type of AI. The one is called as a weak AI and second is called as a strong AI. The one is called as a weak AI and second is called as a strong AI. Okay, weak AI. Very I'm so happy guys. Okay, fine. Around 70 messages, 80 messages I received. Great, great. So happy. Good. 70% people, 80% people, if you understood, so my purpose solved. But I try to connect everyone for 100%. So I am trying, so you people also trying. But one thing I can tell you, like here in this platform, I literally got the people are good. Literally peoples are good. Because many platforms I'm connecting there. But I'm literally telling you that literally peoples are good. They are responding me properly. Their queries and everything have some level. Okay, some level. And just like you people are searching a Dronacharya type of teacher in your class. For your class. Okay. The same we type of teachers also searching the Arjun type of student in our class. Okay. So the things are being mutually. Okay. It's not one-sided. Okay. And I always spoken you guys, my work only for 30 questions. Okay. Your work is for 100 questions. Reason I teach only 2 subjects. That's why my work is only for 30 questions. But your work is 100 questions. A single subject, 1 or 2 subjects will not enough to crack your examinations. So you must have to give a equal weightage to all the subjects. Okay, and those subjects have a more weightage. You should give a more studies and more hard work in that subjects. According to the requirement, you should give your effort. Okay, fine. So you come. So yeah, I was telling you on the basis of performance, accuracy, and reliability, I'm going to check. Literally a machine is working like to the brain 100%. So here 2 types. The one is a weak AI. Weak AI means if a machine is not working like to the human brain, not working like to human brain. If machine is not working like to human brain. If a machine is not working like to human brain. If a machine is not working like to human brain, then that is what? That is a weak AI. Okay, fine. It means a machine is not working better. Okay, in the respect of performance, accuracy, and reliability. Told you guys 100 times I will use this name. Okay, so it's called weak AI. So it means machine efficiency is not like human brain. It's inferior then. It is inferior weak AI. What is strong AI? Strong AI. Working like to human brain. Working like to human brain. It is working like to human brain. It is working like to human brain. The strong AI, if a machine is performing their own work, okay, in the reference of their own performance, accuracy, and reliability, like to the human brain, that's called a strong AI. Now the question is, now here, strong AI. If a machine will work 100% like to the human brain, if a machine work 100% like to human brain, if a machine work 100% like to human brain, then it will be the case of full AI. What is a full AI? If a machine work 100% like to the human brain. See guys, just don't be confused. I'm going to force it. If a machine work like to the human brain, it's a strong AI. 100% like to the human brain. In this way, strong AI is become as a different as a full AI. Then it will be the case of full AI. But one thing I'll tell you, a machine can be like to the human brain, but it's not like 100% work like to the human brain. So practically not possible. Practically not possible. I am not telling you. Scientist communities are saying it. That full AI is practically not possible. But many of the AI you develop, you people have developed, where you will literally get it is better than the human being. But officially, full AI is not possible. So you remember. How a human brain will say about it, he a machine even better than human brain. Then what will be the value of human being? And वैसे भी this machine even developed by human being. You people know very well. Okay, you people know very well. So in that way, guys, even the weak AI, strong AI, you know, all are described. All are described. Okay, everyone, all are described. Now come next. See, some of the example of AI. I think this whole brief wala part you people clear in the beginning, introduction of AI. Introduction of AI, you people very clear. Some of the example of AI I'll give you. Okay, some of the example of AI I will give you. Like, okay, some of the example of AI. College admission test. Very common, I'm telling you. College admission test. One, college admission test. Second one. Okay, one is a college admission test. Next one. Coffee test. Coffee test. Turing test. Turing test is also called as a human machine talk. Turing test is also called as a human machine talk. Turing test is also called as a human machine talk. Okay, so these few examples were right. Turing test also called as a human machine talk. So Turing test is also called as a human machine talk. Okay, so it is also called as a human machine. So what is the human machine talk and all that discussion I will do. Okay, but one thing, and you must be clear here. What is the college admission test? Even guys, if right now have a lot of AI haven't developed. Piyush, that all discussion, you're right, I agreed with you. Okay. Slowly and gradually, whenever this chapter will complete, many of your queries will clear. Because this chapter I will teach in balance. Okay, this chapter I will teach in balance. So wait. Okay, so wait. So coffee test, what is the college admission test? Even I just tell you, you know, last year UPSC happened in October. Okay, this year also is going to happen in October, and you know, in 10 days results came, prelims result out. How so? Through the help of AI. A machine can do scanning of your answer sheet, and just through the help of AI, okay, computer programming, what happened? Your result, your copy will check and your result will come. So your copy check and the result come. It's a part of what? Those copy check and the result come. It's a part of college admission test. Okay, in 10 days, first time in the UPSC history, prelims result came. And this time it also happened. 10th October have your prelims examination. I'm very sure in 2021, 10th October have your prelims examination. Definitely in 10 days your final result will also come. Till 20th, I'm sure. The reason is a machine is working. And see, manually if human being will go and check your prelims copy, 5 lakh people those apply. Is it possible? Is it possible manually? No. And still have some possibility of errors are there. But machine 0% error and all. Here it is good as a AI. It's just a simple examples. So similarly, college admission tests from beginning till last, final admissions, a machine can work in the college and schools or universities. Efficiency better, no partiality. A machine will not take a partiality for anyone, because for machine there is no brother, no sister, no son, no daughter. Okay, what? The system will be very fair. The machine will take a performance, accuracies, reliabilities. Okay, one part. The second one is coffee test. It's very simple and very funny also. Suppose whenever I will come to your house, guys. Definitely when you crack your examinations, I will literally come to your house. It's my nature. We have a best ranking. Okay, best ranking means in top 10. If you'll be there, so definitely I will come to your house, and I will thanks even just to appreciate you and your parents also. Okay. So definitely with sweets and all, and I will also do bhangda whatever. Okay, you people like. But it's also very proud moment for me as your parents. Same me also, and your parents will start to know with your name that day. So definitely you will have a lot of prouds, and in the coming batches I will take your name. So definitely many people say, hey, Ravi sir was taking your class, still sir, Ravi sir is taking your name in the class. So literally you people know it. So definitely in that day when I'll come, so definitely, but purpose is what? Coffee here. Okay, so when I'll come to your house, so definitely you people will prefer me, sir, tea, coffee, anything, because first time you are you prefer or not, don't worry, I just created something that maybe you prefer. Okay, so definitely you will ask me something. A girl's got salad. Then she said, sir, which type of coffee you prefer? Okay, fine. Sir, sugar how much? I said medium. Sir, coffee how much concentrate and what? I said medium. Just listen. What is my medium and what is your medium? Coffee, concentration, everybody understood. So definitely it is different. Definitely it is different. In the case of me, what is the medium sweet? What is you is different. Whatever you people have planned, you just brought it. Suppose I thought it's according to you medium, but it's more sweet. But I will not say anything because you used your love and affection that you have made a coffee for me. So I cannot say, but even I say that really it's very sweet coffee and tea. Okay. So, but ultimately it was something more sweeter. How it is so? The reason is manually everything is going to happen, but in the case of a machine, proper concentration of caffeine, proper sugar content, every value you can set, and according to that value, you know what happened, that proper coffee according your requirement, it will come. Yes, it's what? Coffee test. It's just a simple example I just have given you. Okay, in Pune, you know, next to Mumbai, close to Mumbai. One of the shop is there. Okay, one of the shop is there. Okay. In Pune, one of the shop is there. In Pune, one of the shop is there where the Golgappe are available. Golgappe, I think you people know very well. Pani Puri, many places, different terms is used. What in Bangalore people call is, I think Pani Puri is the term used there. Delhi mein the people use the Pani Puri. Many people use the Golgappe and all. Okay. So a proper Golgappa, how much the sugar, how much the chutney, how much the concentration of salt, how much the spicy, everything you people can plan through the help of AI. Okay, fine. Leave it. Neither you will tell me, sir, morning may you start to say about the food items. Okay, fine. Now you come here, that is something interesting about Turing test. What is a Turing test? Turing test is a human machine talk. Here I will tell you something about it. And literally it is really, you know, it's a great future about Turing test. Okay, what is the Turing test? Human machine talk. Here I will prove, guys. Just see what is the Turing test. Suppose one side, it's a wall. This side me and this side you. It's a wall. This side me and this side you. It's a wall. Okay, fine. You just see. This is the computer. Suppose this is a computer. This is a computer. And a person is here in front of this computer. So it's a computer. Okay, fine. It's a me. Better it's a me. It's me. And this is the computer. It's you. It's you. You, Mr. X, Y, Z. Those are listening my classes. Right now it's you. Okay, fine. Nobody can see to each other. Whatever the query I have on from my computer, I will ask you, you will reply me. Whatever the query I have, you will reply me. Okay, everyone? You will reply me. Now what will come there? And second side. What will come? Second side. Second side is a machine. And this machine is totally programmed by human being. This machine is totally programmed. Okay, this machine is totally programmed by human being. Okay, everyone. Now, how to program, what to program. Just see the result, guys. Okay. I asked also the same question to this machine. Those are also programmed by human being. As an example of AI, let me check. Okay, as an example of AI, let me check. Okay, now listen, what will be here in this case? Okay, what will be here in this case? If I ask the questions to you people, listen, is a Turing test. Okay, I asked a very simple question: a plus b whole square is equal to what? I just mentioned on my computer is equal to a square plus b square plus 2ab. I just wanted to check this question answer from you. You also respond through computer: a square plus b square plus 2ab. You have given me the answer this. And when I ask this question to this machine, you just trust me guys, this machine also has given me the response: a square plus b square plus 2ab. It means those a machine is giving a response, same response, those a person is giving a response, same type of response a machine is giving. It means this machine is thinking like to the human brain. It means this machine is passed in the Turing test. Turing test clear or not? Human machine talk is possible or not? As just a human is giving a response, the same response is machine is giving it. So this machine and this human have a same connections. Accordingly, this machine is programmed, and now this machine can be used to talk with the human machine talk. 100% this machine pass in human machine talk. This is called as Turing test. Clear guys? Turing test. I want everyone answer one. I want everyone answer one. Turing test pass. I want everyone answer one. Everyone answer as one. Katana, you just come. Definitely I can agree. The Katana, that window 10 maybe are using it. Okay. Okay, so Turing test is what? It's a test guys where a machine has been developed that will talk like to the human brain. Okay, if you are not getting understood what happened here, so you just remember, I just has given you a picture, idea how this Turing test even checked. But you people don't go how to Turing test is checked, because this type of answer you will never write. Actually what is the Turing test is the human machine talk, that answer and logic you people very clear. Okay, one thing more, even this type of human machine can be used to check your mains copy. So in the coming future, maybe this type of methodology we will use to check your UPSC mains copy. How it is possible? How it is possible? Very simple guys. Now suppose I am going to check your copy of GS paper 3, and I am giving this marks around 111 marks out of 250. Okay, 250. These are my marks, those I have given you. Fine. And the second side, one more information I'll tell you. What is here the next? A machine, this machine, those I program. It is also check a copy and has given 110, या 111, या 112 marks. So similarly, I can tell you guys, this machine is also giving the same marks as I have given you. Only this machine is programmed. Okay, अब this type of machine can use to check your copy. अब a human being can be biased for some person. I told you guys, for some family member, some whatever the friend circle and all. But this machine cannot be biased for anyone. I am just giving you a future of this Turing test. I am not telling you it is being started. I am telling you there is a possibility in the coming future a machine can be used for these purposes. And how this machine will have a knowledge through the help of internet and many sources. But one thing I'll tell you guys, you know what happened? Whenever we people make a paper, okay, I'll tell you, we will also provide some model answers. Trust me, because I made some question papers, but not for UPSC, not state pieces and all. I cannot disclose. It's not, I should not discuss it. But I'm telling you, whenever the question papers we people are making, we are also providing their own model answers. And there are some keywords also. Okay, some keywords also. Okay, keywords also. Definitely one day I'll get opportunity to make UPSC paper, though that may be also there. So that is we are providing. And these keywords, those people are going to check your copy. Will read first. And later on start to check it. And then parameters are defined. This type of keywords are this is, so this much of marks you should have to give. So this much of discipline we are maintaining to check your mains copy. Many people have started to give me the example. Example wala part application mein I'll go there. First of all, the concept wala part you try to understand. Okay. So it's everything about the future of Turing, just those I told you. I'm now coming to the various discipline. Even driverless car, driverless drone. These are also being a part of what AI. Okay, these are also the part of AI. Like UAE has announced to United Arab Emirates had announced that very soon I'll provide the driverless drone to ferrying in the sky, guys, to carry the people from one place to another place. So maybe possible, and that definitely will work through the help of AI. Okay, that will definitely work through AI. So that will be the part of AI examples. Okay. Actually, many thrust areas of AI will go there. So many thrust areas of AI. Many thrust area of AI. So in various areas where the AI is developed. So different thrust areas of AI. So area where the AI is. So robots is also the one of the part. And here the example is a machine learning. Here the example is machine learning. Okay, here the example is machine learning. Here the example is autonomous system. Here's an autonomous system. The next one is an autonomous system. Third one is a language processing. Third is the language processing. These are the example I'm giving you. Language processing. Even in 2019 when this question was in your essay paper na, so you must have to write entire concepts, but as a story type. Okay, language processing. As a robotics. The one is a robotics. And one more is the artificial creativity. Artificial creativity. Artificial creativity. These are the different thrust areas of AI. Robotics I'll discuss in detail in the later one. After completion of AI, I'll go to the next chapter, that will be robotics. So these all names you should write: machine learning, autonomous system, language processing, robotics, and artificial creativity. You know, machine learning is what? In this case of machine learning, you know what happened? Without being programmed, a machine learn itself. A machine learn itself. Without being programmed, a machine learn itself. Machine learning. Okay, autonomous system. Autonomous system. Autonomous system is any task a machine is going to perform. A system that learn on its own to perform various tasks. A system learn its own and perform various tasks. And perform various tasks. Language processing, you know very well. Language conversions is the best answer. Language conversion. Language conversion. Robotics as a separate I'll discuss with you. Artificial creativity. It's a creativity of a computer. What is the creativity of a computer? Here one example I'll give you, that is called IVA. Is a best example, and this example I will prove how it's happened. Don't think so. Blockchain is a part of AI. Blockchain, you asked me into the miscellaneous chapter. Don't think so. Blockchain is a part of AI. So don't think so. Blockchain is a part of AI. Now you just listen, what are all these thrust areas of AI? Apple, Siri, Flip, Alexa. Okay, many discussion I'll do. You just wait. Google Assistants, many of the application I'll do. But first of all, you just let me finish the thrust area of AI. Machine learning. Even in this case, you people have developed a machine first. And this machine will take various tasks. This machine will take various tasks, but you people have not programmed this machine. But one thing I'll tell you guys, this type of machine learning not provide a better results with more better accuracy? ML, that is called machine learning. Reason. In the case of machine learning, those information we people are going to set in this machine, this information is only few, and on the basis of few information this machine itself work. I'll give you one example. You just tell me, guys, when you people born, from that day till date, what you people got? You people got a lot of informations inside your brain. Your parents told you this is a black color. Your teachers told you it's a black color, and finally your brain said it is a black color. And then at the age of 5 years, when you all people met together, and everybody introduced this color, so everybody say it's a black color. It means your brain is from the beginning is going to program from your house, then your teachers, then your friends, then your books and all. They totally program this is what, and everybody is saying this is the black color. Similarly, I'm telling you guys, those machines are working. The few of the applications we are providing in these machines, and on the basis of these applications, all machine itself being programmed and take their own actions. If this type of AI develop, it will be the part of machine learning. Autonomous system. Here machine itself going to take and solve any task. Autonomous system. A machine is also going to take and solve that task. It's called autonomous system. Just like what about the various conditions of environmental pollutions, and how machine can work, like autonomous system. I can tell you, driverless car is the best example. Various different tasks are coming in front of you when you people are moving your vehicles. Suddenly vehicles are coming towards you, your vehicle is speed decreased. Suddenly front side of vehicle stop, your vehicle also stop and make a distance of 7 to 10 meter. Okay, many applications, many purposes. Okay, autonomous system. Language processing, so you can go through on Google also. English to Hindi, Hindi to English, English to Kannada, Kannada to Telugu, Telugu to Tamil. You can do it very easily. Even one of my friend is Portuguese and does not know about in English. So I just translate the English to Portuguese. And I don't know about the Portuguese. So what she did, even she convert the Portuguese to English, and then we people talk to each other. And how it is so? Because of language conversion. So language conversion is what? It's a process. Through that, how smartly this type of platforms are doing it. Robotics, again I repeat, artificial creativity is a creativity of computer. AIVA is the best example. Acha, just I'll give you one example of AIVA. Even in this period of time, those classes I am taking, it's not working through AIVA. But I'll tell you guys how I can use the AIVA. Okay, suppose front side have a camera. Okay, front side have a camera. Definitely front side have a camera. And suppose that is the recorder. It's not actually recorder. Okay, suppose it's a recorder. Okay, whatever I'm telling you, that every recording is here. And what about the picture and video movement, everything, that recording is there. Okay, fine. Even in this class, in this live class, everything's happened parallelly. This is used to do the recording in my computer desktop. Desktop is here for the all software and everything. Camera is there, sound system is here, and chatting laptop is here. So how this entire system is working in my room, I hope today you understood. Okay. So ultimately what is going to happen in this is, if I'm using the AIVA, in the case of AIVA, that video recording will be there, and audio recording, suppose I have a Sony recorder in my pocket. So my voice recording is here in my pocket, and picture recording is there. Even this camera can also do the voice recording, but see, the camera distance is more. And this is the reason have a difficulty. Camera distance is more, that is the reason here have a difficulty. That proper voice should not be in there. Any other surrounding voice if coming, so definitely it will also be cached there. So finally you have a disturbances. Here 100% the wire is here. Nearby sound still will not work because this is very close to my lips. Okay everyone, fine. So what will come in the AIVA system? That picture recording there, audio recording here. At the last, when the whole class will finished, now what I will do? This both to picture and this audio recording I will club together. through the help of Aiva. And how Aiva is smartly club this my audio and the video on the basis of my lip sync. Suppose I am speaking lip sync. So what about my pronunciations? Lip sync. Some movement of the lips happen. Okay. And accordingly the movement of the lips, all the voice will connect together. How smartly it is. Okay. So I hope I discuss with you guys how through the help of lip syncs the words connect through videos. And see, this type of techniques we people are using for the dubbing of videos into English to Hindi to Tamil, Tamil to Hindi, Telugu to Hindi, Malayalam to Hindi. It's become possible. Yes, many actors of the Tamil movies, they are making videos in Hindi. How it is so? Many Hindi actors, they are making videos on Tamil. They still don't know Tamil and Telugu in the frequently. So the voice is going to dub by other person voice. But acting they are doing performing. And how this both to acting and the voice is going to merge through Aiva type of system. Even most of the South Indian videos I have seen Shiva, many of them I have seen it. Still I like to see VIP, VIP 1, VIP 2. I hope you understood many of the videos I have seen. Okay, fine. So I'm just telling you guys these all the videos, movies, but I don't know Tamil and Telugu. Little I understood, but properly frequently it is not possible. Maybe now I start to go to the Bengaluru frequently. So definitely in 2-3 years, because of you all supports, I will learn other languages also. And it's good if you learn, if you know you can speak and talk to the many languages. It's good. Okay, it's good. And I want to do it. So definitely I hope you understood how the many recordings, audio dubbings happen. So somehow it's working through the help of AI. Okay, so artificial creativity, I hope you understood by the computer. Okay, creativity of a computer. And I think Aiva is the best one to understand by everyone. Okay, now come popular AI projects. Popular AI projects are products. Worldwide those develop. Somebody were using about Cortana here, somebody using here about Siri here. So these are the worldwide many AIs products, right? Okay, different AI products, worldwide AI products. So AI products. So AI products are projects. What about you got? So next one is AI products. Okay, some example I'll give you here. The one that is called Google Brain. I don't know practically what is the Google Brain. Okay, I don't know what is the Google Brain practically. Okay, wait. Next one is Adam. Adam I have seen practically, guys. Adam I have seen. So I can simplify the Adams. One video I'll show you about the Adam. Maci. MACI. Amazon using for the safety and security of their own data. Maci. After it you come Siri. Then Google Assistants. Siri. Then Google Assistants. So Siri. Then Google Assistants. Okay, Siri. Then Google Assistants. So Siri, then Google Assistants. Now come Alexa, which is very, very popular. Alexa, it is very, very popular. Okay, next one is Watson. And one more, that is Cortana. Somebody even told me about Cortana. So these are few examples: Google Brain, Adam, Maci, Siri, Google Assistant, Alexa, Watson, and Cortana. So Alexa here, Muskan, I mentioned it. Google Brain is a deep learning AI project of Google. It's a deep learning project of Google company. But one thing, I'm sorry, I don't know how it is working. Only factual data I can give you. Adam I can give you. Adam is also the kind of deep learning project of Microsoft. Here I can show you one video also. Adam video I can show you. Even one of the scientists will show you one of the Adam platform, and here he will show you how this deep learning project is working. Many people will be here in front of the person, where the person was showing the Adam platforms. And just ask to just call a dog and just ask to all the people those who are in front of them. Do you know which dog breed is this? Which dog breed is this? The people were silent there. People were silent there. Okay, because how many dogs breeds and all, how people can identify, and that dog breed was not very common. So it was difficult to find out. Then what he has done in that, I'll show you that video, just 2-3 minutes videos there. He will capture a pics of that dog and just run on the Adam platform. What the Adam platform will do? Adam platform will certify, check it, and finally Adam platform said that's Rhodesian dog something, and the people has given a clapping. So how interesting a deep learning platform, okay, the Microsoft has developed. You can see here, you can see here. The benefit of the digital board, you see here, guys. This is a video, 1 minute 20 seconds, and just you see here, you will get your answer. Just a minute. Is mute, is mute.
To be developed so that we actually have the opportunity to read. I really want to show you a demo that is actually about the lot of focused work we have in Microsoft Research about building computational infrastructure that will enable all those deep learning, machine learning algorithms to be developed. So that we actually have the opportunity to really have the state-of-the-art recognition engines for practically anything. So to help me out, I'm going to have my colleague Johnson to come up to the stage and talk about Johnson.
Thank you, Harry. Today, I'd like to showcase you the app that Javier built by using it on actual live dogs, just to delight me. Cortana, what kind of dog is this?
For me.
Anyone care to guess what breed of dog this is? Ridgeback? Let's see if Cortana agrees with you.
I believe this is a Rhodesian Ridgeback.
Wow.
So I hope you understood. It's,
Thank you.
okay, you go now. So what was here, I think somehow you people clear. Tell me, guys, you people clear with these concepts? Tell me fast, you people clear with this concept? Missy, it's a similar to Google Lens. Okay, fine, it's similar to Google Lens, agreed. But I think through the help of this video, you people understood it's somehow, okay, just deep learning platforms. What about Massey? What about Massey? See guys, Massey. Actually for Google, Amazon. Amazon is using it. It's an intelligence personal assistance. Amazon Security Services. It is an Amazon Security Services. It is an Amazon Security Services. What is it? I will describe you. Actually, you know, with Amazon, many of the companies have a competitions. You people know very well. Amazon in the last moment showed their own prices and all. Some schemes, yeah, something Amazon is going to bring some markets, okay, some advertisement, yeah, some sell, some offer. Those Amazon is providing in the last moment. So that's why all the information of Amazon, because of competitors, Amazon try to hide it. Even the data those Amazon is going to save, it should be more safe and secure. For that purposes, Amazon is using Massey. So Amazon Security Services. Amazon Security Services. What about Siri? One line. Intelligence personal assistance. It is intelligence personal assistant. Personal assistance. Personal assistant. The same Google Assistant is also intelligent personal assistant. This is provided by Apple and this is provided by Android phones. and that you people know very well. Those you people know very well. And these both two you people know very well. Okay, and these both two you people know very well. Okay, these both two you people know very well. What is it? Because you people have either this handset phone, this handset phone, definitely have it in these two. And you people are using it, the Siri and all, you people know very well. Okay, intelligence personal assistant. So I don't like to waste your time. Alexa, I still don't know. I don't think so. You must have to discuss Alexa. Amazon's intelligence personal assistance. Amazon intelligence personal assistance. It's Amazon providing you speaker, internet connections, Bluetooth, many activities you can use through the help of Alexa. Okay, it is able to listen your voice. Okay, even videos, audio, everything. Even if you have a kids at your home, Dadaji type of people, so everybody can enjoy a lot through the help of Alexa. Watson, I don't know what is it, but it's a IBM company made. Watson is a AI product developed by IBM company, just for question answering. So for this type of purposes, IBM has developed a Watson. Okay, Watson. Question answer computer system. So question and answering computer system. Okay, for that purpose. And Cortana Windows 10 is providing you. Okay, and Cortana, I hope many people have Windows 10. Just like those Siri and that work you people are using in your mobile phone. Similarly, through Cortana, that all the works you people can do on your desktop, your laptop, where you install the Windows 10. Okay, where you people install the Windows 10. So it's a Microsoft Intelligent Personal Assistance. It's a Microsoft Intelligence Personal Assistance. So it's a Microsoft Intelligence Personal Assistant. Cortana for HP. Okay, Mishi. It's a simple, guys. It's not very difficult part. Okay, it's not very difficult part. Okay, I hope you people know about the first humanoid robot. Those got citizenship. Okay, the first humanoid robot got citizenship. So first humanoid robot got citizenship. Bixby given by Samsung phone. अच्छा, मुझे नहीं पता था Bixby. So you should also write it Bixby given by Samsung phone. Bixby given by Samsung phone. Okay. So you add on one more. Bixby given by Samsung phone. Others, Saudi Arabia. Acha, I agreed. The first humanoid robot got citizenship. I hope you people know very well. That is Sophia. See, this is only fact and figure based topic. And see, it got the citizenship in 2017. In October 2017, this was the announcement by Saudi Arabia. The first humanoid robot got citizenship. It was Sophia. And who has developed this? Sophia. A Hong Kong company. A Hong Kong company has developed it. That name is what? Hanson Robotics. That name is the Hanson Robotics. A Hong Kong company has developed it. That name is the Hanson Robotics. Okay, Hanson Robotics. And this is also called as a social humanoid robot. This is the first humanoid robot that is called social humanoid robot. This is social humanoid robot. You must be clear. This is social humanoid robot. Okay, it's a social humanoid robot. And you know, first time when it was shown for the human, even appearances, 62 by facial expressions, it has changed. 62 type of facial expressions. So how many types of facial expressions? 62 type of facial expressions it has done at that time. So 62 types of facial expressions. So that time 62 type of facial expressions. Okay, 1 point. 62 facial expression, good. And see, actually, you know, when we people are talking to anyone, even during my teaching, guys, in different mode, my face have a different expressions. Okay, similarly, as when the Sophia even talking to the in front of human beings. Okay, so that finally, you know, whenever I talked, different expression changed. First time on the appearance, you know, when it came to the public, guys, 62 type, as people notified it, facial expression has done. And one thing more, guys, one of the actress face used there, model face you people know, that is called Audrey Hepburn. That is a Audrey Hepburn. Even whenever I have seen, I did not find out that literally she is looking like Audrey Hepburn or not. But officially it is mentioned its face is, you know, Audrey Hepburn. So Audrey Hepburn face is used there. It's a British actress. So Audrey Hepburn. अब देख लो ऐसे ही कुछ सोफिया है. Okay. So I'm not finding like literally looking like Audrey Hepburn or not, but जो भी है whatever. The face of Sophia is, you know, it's like Audrey Hepburn face is used there. And same time 7 siblings of even this Sophia even developed. Siblings is like brothers, sisters, the same. And all the name of siblings are given with the name of scientists. Hams, Ben, Julos, you know, in that type. So at the same time 7 siblings. So at the same time 7 siblings was, you know, developed. So Audrey Hepburn, 7 siblings. She is able to process speech and can have conversations using Google Chrome. Using Google Chrome. You know, Google Chrome. So what about the conversation you people are doing with the Sophia? Sophia have a Google Chrome, everything. And through the help of Google Chrome, it will check through internet and then respond you people, guys. And to make a eye contact. It's a very intelligent robot. Through the help of to make eye contacts. Okay, even the best part of Sophia is, guys, like it can be used for the healthcare, customer services, therapy, educations. Okay, it is a hope so. The best part that I got it, many applications, but the best part as in the case of nursing purposes. Nursing purposes. Help to the elderly age people, yeah, old age people. Okay, it can be a best use in the coming future. Okay, it will be a best use in the coming future. Okay, it will be the best use in the coming future. The nursing vagara mein we can use it. Okay, we can use it. So there is something you people remember. It's a total fact and figures, nothing more here in this case. Okay, fine. Now come into what kind of advancement in India, AI in India. AI in India. So fact and figure based, guys. Artificial intelligence in India. Most of the now conceptual part over. Now in the last 2-3 chapters, those I'll discuss. It's information based. Information based. Subham, Cyberdome project actually not a AI based. Cyberdome was actually for the safety of a computer system in Kerala. It was introduced, and it was a mains question paper in 2018 also. Okay, 18 or 19 this question came, but it's not directly a base of AI. Okay, directly not a base of AI. So AI in India. Okay, India stand the 3rd position in the AI research, and sometimes 4th positions. So India's ranking is either 3rd or 4th in the case of AI research in India. Worldwide AI research in India. Actually, there was a news in 2018. In 2018 budget. In 2018 budget, first time in the history of India. Basically, AI is a new technology. Okay, here in this budget, there was a provision for the AI research. In this 2018 budget, first time there was a provision of AI. And after this budget, guys, what about the various research and everything India will do? Okay, purpose was to work into the machine learning, artificial intelligence, 2018 budgets. And then Niti Aayog report. Niti Aayog even asked to the government, Niti Aayog. Okay, even asked to the government. Niti Aayog asked to the government. Okay, purpose, Niti Aayog asked to the government to make, okay, to make overall, to prepare a roadmap, to prepare a roadmap, to prepare a roadmap. Okay, to prepare a roadmap. So Niti Aayog to prepare a roadmap. Okay, to prepare a roadmap, especially how India will go for the AI work and all. So every roadmap, for that purpose, Niti Aayog just to ask to the government of India for its roadmap. Okay, how AI based technologies and every work we will do here. Okay, one thing more guys, different type of AI startup in India. Different types of AI startup in India, and this is a good question for your mains paper. What kind of advancement in India? Even whenever UPSC asks the questions from robotics and nanotechnology, UPSC asked, what about the India's advancement in the robotics? Nanotechnology based question, those UPSC are in the mains paper. Okay, how it's India's future, 21st century. Nanotechnology is the future, yeah, 21st century technology. In this way, UPSC asks the questions. Okay, so similarly guys, about the AI startup in India, it means in India have a different type of AI based, technologies innovations happen in the various sectors, and that you must know here into the factual data. AI based, the one here I'll tell you that is called active AI. The one is here that is called active AI. The one is here that is called active AI. The second one is a Alendra system. Alendra system. The first I am writing this, and one thing more guys, these all are the factual chapter. So once you must have to mug up it. Other chapters which you people have listened me earlier, 7 chapters, 6, 7 chapter I finished. Okay, that all was the more conceptual questions. Okay, where UPSC make. Here some chapters are factuals. And that is the reason in the last those chapter I brought is a factual, but equal importance. I told you guys, either paper is a conceptual or factual does not matter. Each and every question have a same weightage. Either the question paper is simple or tough, does not matter. Every question should not wrong. Reason every question have a equal weightage. Okay, so you remember. Okay, either the question paper is a simple, advanced, factual, conceptual. You cannot miss any opportunity because every question have a same weightage. Even when I discuss the defense, I'll take around 3 hours. Many discussion I will do. But at last I'll tell you, here I have to discuss entire defense technology part. But defense technology based question generally current affairs based. And then I'll tell you at the last, there is no question in your paper from the defense. Actually, if you see overall in the last 10 years, hardly 4 times in the last 10 years, UPSC made a question from defense technology. But here I'm discussing entire science and technology. So how I can skip defense technology? That's why I will discuss. But the weightage of defense is very less. Even whenever I will discuss the defense technology, I will share a PDF to everyone, and those PDF you people will follow. Okay, those just like in the case of schemes of energy program and all, I have given you the PDF. The same I'll do here in this case. Okay, so through PDF I will go, and finally the 3 hours I will finish the defense. Now the detection part, if you will start, so at least 2-3 days I will waste. And the last you will get it's a question paper, or questions are also very less. But yes, if any kind of current affairs are coming from defense based, on the basis of concept part, you will understand. Oh, this is a factual data. Oh, new IS cavalry is coming under the commission right now. Okay. Please check noise. But yes, if any kind of current affairs are coming from defense based, on the basis of concept part, you will understand. Oh, this is a factual data. Oh, new IS cavalry is coming under the commission right now. Noise problem is not very minute, is there? Please check noise. But yes, if any kind of current affairs are coming from defense, on the basis of concept part, you will understand. Oh, this is a factual data. Oh, new IS cavalry is coming under the commission right now. Hello. Hello. When I get close, the voice problem increase. Okay, but I'm not getting when I'm getting close to the board. Okay, fine. Okay, fine. I think I should start. We have 20-30 minutes more classes. When I get close, the voice. Okay. Come. Some AI startup in India. The Active AI. Okay. Alendra system. The next one is the artificial. Next one is the artificial. Next one is the artificial. Next one is the Artivatic Data Lab. Next one is the Artivatic Data Labs. So next one is the Artivatic Data Labs. Next Arya AI. Next Fluid AI. Next one is Fluid AI. Next Marf AI and SIGTuple. Marf AI and SIGTuple. So these are the many AI startup in India. Okay, many AI startup in India, and I think if mains paper based AI is coming as essay wala question 19, so everything you must have to remember there. Okay, come. What about the Activatic AI? Okay, what is the Activate AI? Again, if any problem happen, so you just write it. Actually, you know what happens, sometimes I forget, I just too busy here, but I don't like to miss your chat, that is the reason. Okay, so I think I should start. Okay, so activate, there's a factual data, you just write it. Activatic AI. Activatic AI is providing the conversational banking services. Conversation banking services. Okay, for conversation banking services, through the help of this feature, you people understood very well. Within a bank, for conversation purposes, this type of AI is being developed. Your queries, your informations, whatever you want, guys. Okay, your bank, your bank balance, everything you can get through the help of it. Okay, Activating AI conversational banking service. It's a factual data, guys, and you must have to mug up it. Okay, you must have to mug up it. 1 point. Second one, Alendra system. Okay, this is the system generally used to identify objects. To identify an objects. This is the AI has been developed to identify an objects. Detect and identify objects. So it will identify any objects, even any peoples. It is also very important for the detection of the particular person. Okay, so authenticity will be there if a person will identify through the help of this platform. Okay, through the help of this platform. Artificial is also the kind of AI. Okay, that is of deep learning platform. Okay, this is of deep learning platform. So artificial is a deep learning platform. Mug up, guys. Here in this case, I request to all of you, mug up it. Only in the brief I'm telling you, I'm telling you what about the feature of it. Artivatic Data Lab is what? It's a very tiny robots. It's very interesting, and even it's literally very useful for the coming future. A tiny robots. It's a kind of tiny robots. Tiny means very small in size. It's a kind of tiny robots. This tiny robots you just implant inside your body. It will go and screen the whole body parts. It will go and screen the whole body parts. Screen whole body parts. It will go and screen the whole body part, one side. So whole entire screening of your body part will be here, in this case, the one benefit. It will screen the whole body parts in the case of TANeLab. And you know, guys, if anywhere, any kind of problem is going to be happen, suppose some cancerous cell or something is going to form, so there is a possibility through the help of screening, it can provide a lot of abnormalities if any abnormalities are going to happen inside your body. One side. Second one, guys, if you have any problem inside your blood vessels, if blood vessels have any problem, your arteries, your veins have some clotting there, suppose. Okay, that's why have in the coming future possibility of heart attack. So you people can also clean those clotting are happen into the blood vessels. So blood vessels clotting, we can also remove through the help of tiny robots. Okay, through the help of tiny robots. Fine. The next one is the ARIA. ARIA is a neural network. It's also very interesting, guys. It's a neural network. It is a neural network. Even one more discussion I will tell you under the miscellaneous topic. Whenever I will discuss about the cyborg. Cyborg is very popular topic. Cybernetic organisms. Here in that time I will discuss with you that is called Neuralink. Neuralink is a computer neuron interface. Similarly here, guys, it's a neural network. Your neurons and computer interface. It's a neuron and computer interface. What is it? Suppose, guys, I am giving you information. Somebody is a paralysis. Yeah, in coma. The person brain is working, but the person cannot convey their own message to anyone. But don't worry, through the help of screen, whatever the person will think, everything will come on the screen, and whatever the person will say, that person can listen. But the response cannot do. I have a question. I hope you people heard the name of Stephen Hawking. Which person has developed a Neuralink type of features? Exactly not Neuralink, but this type of features, especially for him. Anybody can tell me which company has made the brain computer interface for actually Stephen Hawking? He is no more everybody know how the concept of black holes and all descriptions he has given us. And this is all the discussion I have done under the particle physics, guys. Okay, which company has developed it? And similarly here the India has also one neural network, neuron computer interface, RAI. That is called as a RAI. Okay, that is called as a RAI. Fluid AI for facial recognitions. Fluid AI, I need some space. Okay, Fluid AI. Okay, Fluid AI. The next one is the Fluid AI. Fluid AI, not ARIA, Fluid AI. Okay, I think nobody have information about it. So here the answer is, Stephen Hawking, that is a Microsoft had developed. Yes, Microsoft company has developed. Okay, Microsoft company has developed. Okay, Fluid AI, it is used for facial recognitions. Fluid is used for facial recognitions. Facial recognition. And again, it can also use in the banking sector. It can be also used in the banking sector. Okay, it can be also used in the case of banking sector. Okay, it can also use in the banking sector. You must be clear, guys. Banking sector. Okay, many here. Marf AI is very beautiful, guys. Marf AI is a kind of chatbots. I think it is also available on our website. Chatbots. I think it is available. Okay, chatbots. People can do chatting. Even whenever you people go on the inside, I should open up. You just check it. Okay, whenever you people will go on the website, I think it is available. Just pop up, something will open like it. Okay, suppose it's a page. It's a web page on your website. When you people go on the web page on a website, so some of the pop up something open here automatically. It's looking like something is going to type here. And then said welcome to Insight World. Insight on India. Okay, welcome to Insight on India. Okay, fine, Bangalore. I'm now just, yes, I have a query. I want to join a full GS course. Suppose I want to join full GS course. I'm now then asked which time you want the live class. See, ultimately it is looking like that some of the person is in the background is responding you, but it's not a person. Automatically everything is going to be happen. Okay, it's a chatbots. You know, Ask Disha. I hope you heard it. Ask Disha. This is also a chatbot. Okay, earlier it was available in English, but now it is also available in other languages. Okay, Ask Disha, and this is for railway inquiry. IRCTC brought it. It was in the last 2-3 years in the current affair news. Ask Disha. I'm telling you, AI startup in India, so definitely I should use the name Ask Disha. So Ask Disha is a name of chatbots. Okay, everyone, it is here. Ask Disha. Okay, Ask Disha. AI startup in India, so definitely I should use the name Ask Disha. Okay, Ask Disha. So this is the about the chatbots. I think you will remember it. Okay, this is also the kind of AI. Next one is SigTuple. In the medical diagnosis purposes, that will be a best results in medical diagnosis. In medical diagnosis, it will be a best AI startup in India in the medical diagnosis and all. Okay, so AI powered visual analysis of medical data. Actually, you know, guys, lot of the medical data, okay, people have, and it is not easy to collect and store somewhere. So maybe in this case, SigTuple will give you a better platform. Sometime doctor even asks you, any problem happened, you have earlier. We don't know 15 years back that you have also have some problem in this part. There's no report, nothing as there. So somehow this type of platform will help it. What is the question, Nasasa? Hike. Okay, maybe something people are discussing. Okay, fine, whatever you are doing. So I think you people comfortable, huh? Shamu, it's good. And for the robotic surgery, I'll discuss with you Da Vinci surgical system. I agree. Da Vinci surgical system. Okay, but that part I'll discuss with you. And this Da Vinci surgical system, we can operate through the help of US based doctor can do the operation in India through the help of robotic surgery device. But here not. I included this topic into next chapter, robotics wala. Okay, Da Vinci surgical system. It's a robotic surgery device. Da Vinci surgical system. And this type of system we can use for the telesurgery purposes. Telesurgery. Tele means distant surgery. What is it? I'll discuss. Wait. Under the robotics, that is a part of telemedicine. Telemedicine have a radiocardiology, teleradiology, telesurgery. There are many dimensions. Many dimensions are there. So wait. It will be there in the robotics. Just after it, I will finish. AIMS doctor you are doing. AIMS doc have done it. Samu just hold bacha 2010 mein the first time this operation was happened in India against myasthenia gravis disease where the thymus gland was removed outside. Thymectomy was the issues. Muscular disease was there that can damage the whole body. So you just wait, I'll discuss. Acha, sometime I think so people are showing themselves, okay sir, I'm also very intelligent. Okay, good. When I said you people are not intelligent, definitely you people are good. You think over the UPSC examinations, it also showing that you people are literally, you are good among all. Neither you will think about UPSC. So to think about UPSC is big. It's showing that you have a big. Okay, fine. So wait. But that discussion I'll do into the robotics, not here. Okay, into the robotics, not here. Now come next about advantages and disadvantages. Actually, you know guys, DRDO, in the reference of India, I'll tell you, DRDO. One of the lab of DRDO is called CARE. Okay, CARE. CAIR, CARE. Center for Artificial Intelligence and Robotics. Center for Artificial Intelligence. Artificial Intelligence and Robotics. Because this time I'm discussing about the AI startup in India. So I must have to use the CARE. Center for Artificial Intelligence and Robotics. This is one of the lab working under DRDO. So under DRDO it is working. So definitely this type of lab will start to develop different type of AI. Different types of AI, those we can use into the defense sector. Okay, different type of AI used in the defense sector. So it means DRDO based one of the labs still working to involve into the different type of AI that will improve our defense sector. So this is here. That will improve our defense sector. Center for Artificial Intelligence and Robotics. So Center for Artificial Intelligence and Robotics. Okay, now come into the advantages of artificial intelligence. Advantages and disadvantages last. Advantages of AI. And then next we'll go into the disadvantages of AI. So advantages of AI and disadvantages of AI. So come into advantages and disadvantages of AI. So advantages of AI and disadvantages of AI. Okay. See, advantage of AI, guys. If a machine is taking a decision. Acha, one thing more. Those advantages and disadvantages of AI, I am telling you now, the same will be the advantage and disadvantage of robotics. Because robotic is one of the applied AI. Okay, fine. Those discussion I'll do then next. So this discussion again I will not do in the case of robotics. Okay, so what is here, guys? If a machine is taking a role, so definitely complex and stress worked can be easily performed. Complex and stress work this machine can easily perform. Nobody have any doubts? Any complex and stress work this machine can easily perform. Okay, and see another one, guys. Machine perform work sooner than human. Sooner than human. So time period, the machine will perform the work as soon as possible because machine not tired, everybody know. Okay, machine not tired, everybody know. Okay, no injuries of human, guys. No injuries of human. Injuries human. So no injuries to human. And no even matter of human life. So any difficult task, where have a risk, guys? Where have a risk, guys? You can replace the human through the help of this machine. Advantages of AI. And see, whenever the machine is doing the work, less error will be there. Less error will be there. Less error and defects. It's a benefit when the machine is going to perform. So less error and defects are there. So less error and defects are there. And another one, guys, minimum resources use. Minimize resources. Minimize resources. Minimize resources. Those input resources are required here. It will be very, very less. Okay, minimize resources. So there are many advantages. I hope you people can see here, guys. Many advantages here you can see. Okay, many advantages here you people can see. Minimize resources and all. What about the disadvantage of AI? See, disadvantage of AI, guys, definitely, guys, if a machine is start to take a role, so lack of human sensation. Okay, because machine have no sensation, lack of human sensation. Machine do their own role professionally, but human being can do their own role as a emotionally or something other. Okay, fine. So lack of human sensation, I'm telling you the one, guys. Okay, next. Corruption increases. Why corruption? The people will fire from the job. Even here I agreed with. Because the people does not like to do work. That is the reason, guys, government sector is not working and the private sector is growing. The reason you people know very well. Even in the private sector, we all people even very, very professional and we come on the time and they stop on the time. These classes, I'm giving you one example, but government may you go and see the teachers. They even not do the updations. They are not doing the study even. Whatever they read in their own BA, MA or BSc, MSc or BTech, MTech, they are coming through that and they are discussing it. They still not update. But how much the updation is required in UPSC? They never enter into the class on the proper time and they left the class before the timing. Okay, so that type of thing. But here a machine will perform everything task properly. Okay, and that is the reason the machine is taking the place of human being. Obsession, sensation, whatever, it is another part. But what about the disadvantage? I hope you know very well. The new generation will go towards more corrupt and something will not have a job. Okay, in that way you can call it. So replace the human jobs. So human capabilities are not replaced it. Feeling, willing, there is nothing here in this case. Okay, in that cases. And see, no even one thing more, guys. Even that some some error happen in the machines. Okay, yeah, programming those happen, it will misprogram. Okay, yeah, not proper program there, guys. So it may also be react as a, many movies you have seen, robots, Ravan. So this type of possibilities are also there in the case of disadvantages. So public sector units working hours are more than the private sectors and doing lot of work too. Maybe by public sector units are working, maybe they are doing it. But those public sectors I have seen in my life, banking में तो मुझे दिख रहा है क्या हो रहा है. Heidel के officers में I know very well, the people are not giving the respect. They were not talking properly to you people, guys. Arrest you people now. You people don't take it on yourself. Okay, sir, I am a government teacher, I am much more good. I am not saying exceptions everywhere. Okay, exceptions everywhere. You don't feel ki I just said to you and hit to you. No. I just said about maximum is what majority of them. It is not fine to say that all IAS officers are corrupt. No. Many of the IAS officers still they are very honest. Khemka and many of the IAS officers, there are examples. Okay, fine. So that, but literally government is what you people know very well. Acha, you don't go on that discussion. We are on the science and tech. Okay, I'm sorry if I said and anybody's got hurt because of my words. Okay, fine. Now come robotics. We have a 5 minutes, 10 minutes. I can introduce the robotics today. Okay, fine. Robotics. Actually, robots came from the Greek word. One thing I hope you remember very well. It is one of the example of AI. Robotics is one of the example of AI. Okay, robotics came from a Greek word. The name of Greek word is called as a roboto. That is a roboto. Robotics came from a Greek word roboto, and the meaning of roboto is what? Self-labor. The meaning of roboto is called self-labor. So meaning of roboto is what? This is a self-labor. So meaning of roboto is called this is a self-labor. So robotics came from Greek word, means self-labor. Okay, means called as a self-labor. So means a machine is going to perform their own role, self-labor. Okay, and how a machine perform the role? Through the help of their own programming. Through the help of their own programming. And the name of program, those we are doing in the robotics, term is used. This term you people remember, this is called automations. This is called as automation. So automation is the programming in the robots, those we are doing, and accordingly this robot perform their own role. So automation is the kind of programming those robots is doing. On that basis robot perform. Robots on the basis of performance, accuracy and reliability. Again I am using the words. Robots on the basis of performance, accuracy and reliability. Okay, fine. There are two types of robots. The one that is called as a domestic robot. The one is called as a domestic robot, and second is called as an industrial robot. So one is a domestic robot and second is an industrial robot. Those comparison here I am doing on the basis of performance. Even the performance are robotics. Here the center will be human. So the comparison of a machine with the human I will do, and then I will divide the robotics into two types. The one is domestic and second is industrial. Domestic robots generally not better in performance. Not better than humans. So they are not better in human beings. Again, in which respect? Performance, accuracy and reliability. But industrial robots are defined. Okay, their call is they perform better. They perform better than human beings. They perform better than human beings. So industrial robots, they perform better than human beings. Okay, they perform better than human beings. Here I can give you examples where a machine perform better than human beings. Okay, here I can give you an example, better example where a machine perform better than human beings. Generally, industrial robots you can see in better you just imagine, guys. car manufacturing industry. Car manufacturing industry. Car manufacturing industry. Packaging industries. Lot of packaging industries. Packaging industries. Electronic industries. Suppose somewhere we are going to develop the PCBs, printed circuit boards. Electronic industries. Actually, those car manufacturing industry. Okay, near to my place is the Gurgaon. Which is the name I don't know. Gurgaon is, I hope you people know this place where the International Solar Lines headquarters we are going to set up. Okay, have a Manesar is nearby. Is a Maruti car industry is there, and those industries are there, guys. Thousands and thousands, more than that, guys. I don't know exact number. Cars are getting ready every day on the basis of requirement. So this multiple number of cars are get to ready in that industry. It's what industrial robots. Here the people not involved manually, guys, and that is the reason performance accuracy. Even human being can do a small error in the vehicles, but the machine will not do. Okay, so how properly a machine is going to sit and make a cars and everything. Okay, it's the best example you can see here. It's the best example you can see here. It's a car manufacturing industry. Packaging industry, millions and millions of the bottles, Pepsi and Cola's bottle is getting ready every day. Lot of the cartoon packaging and all people are doing every day. Massive industries. Printed circuit boards, PCBs, those we are using in the electronics. Million, trillion PCBs are getting ready in the industry every day. Not even the small errors are there. These all are the types of industrial. Okay, so these all are the example of industrial robots. So in this way, guys, have a domestic robots, cleaning of your house. To see about you people, guys, like any unwanted person is coming, enter inside your house or not. Intrusions of the person inside your house, it can check properly. Domestic, cleaning of house, intrusion of person. Okay, in this way, domestic robots are working. Okay, in this way, domestic robots are working. I hope you guys clear. Okay, and now later on we'll go into the history of robotics. Then component of robotics, and then we'll go into the application of robotics. After application of robotics, achievements in India, and its future of robotics in this way. Okay, and definitely, guys, achievements and future in the reference of India. Last, I think, with this connection about the advancement of robotics, that was a UPSC question in 2015-16. Advancement in the robotics. So all dimensions, different dimension will connect here. Okay, so all different dimensions will connect here. Okay, history of robotics, components of robotics, application. Again, I'm telling you guys, this topic somehow you will get factual. But the next topic, nanotechnology. Literally, you people enjoy a lot. And this year, CNT was a UPSC questions, 2020 prelims. Okay, everyone. So it is the entire thing. So whatever the discussion for today, it's finished. And 3-4 days more, I'll use these classes, and then my class will over. Okay. So thank you, everyone, to giving me the proper time and to make my class even more comfortable. And 15 minutes break, as every day I take 5 minutes or 10 minutes, but I have given the 15 minutes. I think it was better and you people comfortable. So accordingly, you people plan for the coming 3 days, 4 days more, my syllabus will over. And so thank you, everybody. I hope now if you people have any queries, so you can ask me. Okay, and then I will leave these classes. But I think so query should be very less, because at the time of discussion, whenever any questions are coming, so I discuss with you. So tell me, guys, whatever the discussion I have done, the remaining part of biotechnology and AI, I hope you people comfortable it. And the essay wala topic, that was a UPSC as in mains essay 2019, artificial intelligence. So you have a complete dimensions, ki how much the dimension you can connect through artificial intelligence. So that's why I started artificial intelligence as a story, a machine to have work like to the human brain in this way. How it work, what is it, Okay, in that way I have discussed it. Anybody can tell me who is the father of AI? Anyone? Who is the father of AI? Last. Okay. Which man is called as the father of AI? Anyone know? Because everything I have discussed, but then that name of person I missed. Who is the father of AI? Thank you, Chaman. Thank you, Bacha. Thank you. Who is the father of AI? Last. Father of AI. Anyone? People started to Google, huh? Google. I know you people are very smart. Okay. Cute, very cute. Okay. So, thank you, Chaman. Thank you. Thank you, everyone. Thank you, Prabhakar. Thank you. Mains practice questions. Okay. Fine. McCarthy. You right. One more person, Akshay, is there. Okay. McCarthy. And one more person is there. One more person is there. McCarthy. One more person is there. And Marvin Minsky. You remember these names. Okay. These people are the father of AI. Okay. Father of AI. Shubham, don't worry. I could not discuss the cyber security here, but I promised in the last day, don't forget, it's your duty, okay, to ask me cryptocurrency and blockchain. Don't worry, it's very easy topic, and I'll take around 20 minutes, 30 minutes lecture on the blockchain and the bitcoins, and trust me, then you will never forget in your life if any questions, any queries you have. You people have a trust, you people have a trust on me. So I am telling you, just you people have shown me your interest and the trust, love, affection, care, everything in this 20 days. Okay, I think 20 days over. The 4 or 5 days left here. You just trust me, it will complete. And then you will never forget it's my words. Don't worry. Okay, Shubham. So I think you people can go right now and whatever you do. So thank you, everyone. Okay, trust on me. Go. Bye. Take care. I'm happy that you people are crazy to learn with me. I'm so happy. And you people have a right on me. I have given this right to you. Okay, ask me. I'm happy to know it, guys. Like literally, students want to learn me. I want to be teacher that I can solve your all the problems. Okay, not to create the problem. So don't worry. I'll do. Okay, bye-bye. Good day. Bye-bye.